A big hello to all my PU2 aspirants. I hope you are doing wonderful, my dear little children. Well, I welcome you all to study the passing package questions. So over here, I have packed it with so many questions which are really important for you because they have been repeated so many times. And God knows, this time also it could be repeated. So I just want one thing from you, that is, stay focused on biology while we are watching this particular video. Or basically, you are watching this particular video. And you have to be sure that I am going to do all the questions that ma'am is telling over here. I'm not going to skip even a single question, and I'm going to repeatedly solve it throughout because it will be really helpful for you, my dear children. That's why I'm telling. So I have repeated this question so many times in 2 marker, 3 marker, 5 marker marathon series, but still I'm here. Why? Because these are the most repeated and the most important questions that could be coming throughout your way. So there's a smart plan to it. Okay, you just don't have to randomly study anything. The smart plan. What is a smart plan? You see a number of chapters over here: sexual reproduction and flowering plants, human reproduction, reproductive health, principles of inheritance, molecular basis of inheritance, evolution, human health and diseases, microbes and human welfare, ecosystem. There are certain chapters. It's not 8, it's 9 of them over here, and I call them magical 9 chapters. Why do I say so? Because if you study these chapters, okay, you are going to get 60 plus above for sure. Every single question, if you attempt from this particular chapter, my dear children, if you are aware of this chapter really well, you are going to score 60 plus marks from this particular thing itself. Okay, how, you may ask? I'll tell you. So basically, talking about Part A. Part A consists of MCQ questions, my dear children. In MCQ, you have to attempt almost 15 of them. Almost 15 of them. So over here, the 1 marker questions that these chapters are going to give you are 1 plus 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So you are going to attempt 13 MCQ if you study these chapters. Okay, now 13 marks is sure. Now going ahead with 2 marker. In 2 marker, in your question paper, you have to attempt only 5 2 marker. The options could be many, okay, but you have to attempt only 5 of them. So any 5 you pick: 1, 2, 3, 4 and 5. Okay, so 5 into 2 basically 10 marks. 5 into 2 is equals to 10 marks. So 13 plus 10, it is obviously 23 now. Okay, going ahead. In 3 marker, again you have to attempt 5. So from here only you can attempt the 5 questions. So 1, 2, 3, 4 and 5 I attempted. So how much marks, my dear children? 5 into 3, basically 15. So 23 plus 15 is equals to what? 38. Agreed? Now going ahead. Talking about the 5 marker questions. So in part D, my dear children, this is part A, this is part B, part C, and now part D have the 5 marker questions. In part D, there is 2 sections. In section 1, you have to attempt 4 5 marker. In section 2, you have to attempt 1 5 marker. So 4 plus 1, 5 marker questions you have to attempt. So suppose I attended 2 from this, 2 from this and 1 from this. So 5 into 5 is equals to what? 25. So 38 plus 25 is equals to what? You get your thing as 63 marks. You get 63 marks from this only. So this is passing package. These chapters that you see, this is your passing package. You would definitely pass, and not just pass, you're going to get 63 marks out of it. Now calculation error could be there here and there. Please forgive me if there is. Okay. So but yes, that is what I was telling you. So what you need to do is you have to focus on these chapters. Why I'm telling you have to focus on these chapters? Because it's a mixture of easy and difficult chapters. So over here, you have sexual reproduction, flowering plants, bit lengthy, but okay. Principles of inheritance, again bit lengthy. Molecular basis of inheritance, little lengthy. Human health and diseases, again little lengthy, and evolution as well. Now if you talk about human reproduction and reproductive health, it is kind of bit medium difficult. It's not very difficult, right? And then microbes and human welfare, if you know, remember the name of microbes, it becomes very easy. Same with ecosystem as well. It is not very difficult. So you might feel human reproduction is also difficult. Take it difficult. It's fine. But you have reproductive health. That is a chunnu chapter, right? That's a very small chapter. Now you have microbes in human welfare and ecosystem, again a small chapter, not very big. Okay, I agree the names of the bacteria is difficult to remember, but if you do so, don't you think you can score really well? Because the straight questions are going to come. You're not having to explain a lot of things over there. Straight questions are there. If you remember certain things like that, you're going to score good. So we are going to focus on these chapters first, and then we will take up other chapters. Got it? Works for you? Chalo. So these are the things according to model paper 2 also. This is the entire thing. Model paper 3 blueprint also. This is the thing. So anyways, whatever you do, I have did the calculation really well, and then I have come up with these chapters. You might be asking me, why are you not picking other easy chapters? No, if you pick other easy chapters, you would not be able to score, because I have to see whether these are going to give you 5 3 marker and 2 marker and everything. You get it? So that is the thing, which is why I have chosen these chapters. And now getting ahead with the first chapter that we have, sexual reproduction flowering plants. It carries 11 marks in your exam, my dear children. So over here, the first question, complete the tabular column given below with respect to male gametophyte of angiosperm. So you should be knowing about the pollen grains, my dear children. In pollen grains, over here, you're asked, what is the cells of the male gametophyte? That is, what is the cell of the pollen grain? So shape of the nucleus of the cell. You should know the male gametophyte cell, that is vegetative cell and generative cell. You should know the shape of the nucleus of the cell. The vegetative cell is what? Basically having irregularly shaped nucleus. The generative cell which is there, it is having spindle shaped nucleus. So all this information is really important. That is, you get 2 marks out of it. Now going ahead with the next question. Differentiate between geitonogamy and xenogamy. So again, you should be writing the definition of geitonogamy first. Okay, again you wrote definition of xenogamy also here, and then you tell which one will bring out more variation. Okay, so geitonogamy is happening in the same plant, is happening in between the different flowers of the same plant. So genetically it will be similar to the parent plant. The offspring that will be produced will be similar. Why? Because the pollen grain is from the same plant, the stigma is also from the same plant. So what do you expect? Now xenogamy is basically, it is giving rise to genetically different varieties of plant. Why? Because you have involved 2 different separate plants. It's not same plant, it's 2 separate plants. So these are different. If you write 2 points properly, you get 2 marks each. You got it? Write 2 points properly, not like this in tabular form. You have to write it properly. Cool. So this is one very important question. Next is, what is parthenocarpy? Give an example. Again, you have to write the definition. What is parthenocarpy, my dear children? Where the ovary, which usually becomes the fruit, okay, after fertilization, is now not needing fertilization. It says without fertilization also I will become a fruit. This is parthenocarpy. How does this happen? Because of hormonal changes in the plants, my dear children. And we can induce this also. We can put certain hormones and we can induce it, okay, unnaturally as well. An example is banana. Example is banana. So this is how you have to write the answer. These are all 2 marker questions. Now define this following terms: staminate flowers and pistillate flowers. What do you mean by staminate flowers? Staminate flowers as in which has the stamen, just the stamen. That is, it just has the male reproductive part. Pistillate as in the one that has only the female reproductive part, that is the pistil. You get it? So this is how you need to write the answer. Again, A, staminate flower, underline it and write the definition. B, pistillate flower, underline it and then the definition. Children, I am telling you, I am not here to explain you the answers, because if I do so, it will be really long. So I have explained all this in 2 marker and 3 marker. So wherever you see 2 marks, that means I have explained it in 2 marker, 3 marker marathon series. Okay, and whenever you see 5 marker, that means I have explained in 5 marker marathon series. It is there. I have explained each and every question, every single question. So please watch that video if you are looking forward for explanation, or if you're just looking forward for your questions, you have already studied a bit of thing, then I'll recommend you to go ahead. Okay. Now, sporopollen is one of the most resistant organic materials known. This is a very important question. It comes in the form of MCQ also. So you have to talk about sporopollen and why, how is this resistant? Resistant to what? Strong acid, strong alkali, everything. And why is it known as resistant? Because no enzyme can basically break it down. And where do you find it? It is found in the exine, the outer layer of the pollen grains. So these 3 points should be there on your paper as well. Okay. So basically, what are they resistant to? Why do you call them resistant? Because no enzyme can work on it. And where is it found? Okay, cool. Next, differentiate between pericarp and perisperm. Pericarp is what? The covering of the ovary, my dear children. Pericarp is the wall of the ovary that will develop into the wall of the fruit. So in a pericarp, you have 3 different things: epicarp, the outermost thing; endocarp, the innermost thing; and mesocarp, the middle thing that is there. Mesocarp is usually fleshy. In some fruits, it is not fleshy. Now, perisperm, what is it? So in some plants, usually what happens is what? The cells of the nucellus which is there, it is getting used up. But in some plants, the nucellus still persists and provides nourishment to the growing embryo, my dear children. In that case, you will call this persistent nucellus as what? Perisperm. So perisperm is different. You have to remember the examples over here. So you have 2 examples and they are what? Very important example. Black pepper and beet. Why do I say this is very important? Because it can come in the form of MCQ as well. Okay, cool. Now going ahead. Draw a LS of grass embryo diagram and label the following. So this is a very important diagram under the diagram section, one of the most important diagram every time it comes. So LS of grass embryo, as in you are talking about monocot embryo. This is the monocot embryo. You should be knowing that this thing that you see is the cotyledon, and you call this cotyledon as what? Scutellum, my dear children. Then you have shoot apex. Shoot apex is covered by something that is known as coleoptile. You have root apex, that is radicle, which is covered by root cap and also coleorhiza. Get it? So all this information has been put up over here. You have to label it, you have to draw it properly. So make sure you are drawing it. So this is the LS of grass embryo, very important diagram from this particular chapter. Going ahead to the next question, that is draw a label diagram of transverse section of young anther. Again important. Okay. Why? There are only 7 or 8 most important diagrams which are being asked in the model question paper this time. So the ones that are there in the model question paper, can you skip it? No, never. So basically over here, the transverse section of anther, as in you have to mention what is porogenous tissue, what is it covered by, tapetum it is covered by, outside the tapetum what is there, middle layer is there, outside that endothecium, outside that epidermis. So all 3 things. So all these things should be mentioned. This is the enlarged version. You just have to draw this and label it properly. So this is a transverse section of young anther. So basically if you cut like this and see like this, that's what you find. Cool. Now going ahead, write any 4 characteristics of flowers pollinated by wind. Now this is a 3-marker question, my dear children. So from starting the diagram itself, they are all 3-marker questions. Okay, make sure you are keeping a note on that. Now write any 4 characteristics of flowers pollinated by wind. By wind. So what are the characteristics? So I'll tell you. These flowers, basically you have to make sure the pollen grain is not sticky. If the pollen grain is sticky, my dear children, what will happen? It will go stick there. It has to move around. It has to be light weighted and it has to move around really well. Talking about the stigma which is there, my dear children, it should be feathery. Okay, it should be feathery. Why it should be feathery? Because it has to trap down certain things. Okay, it has to trap down the pollen grains which are basically flying in the wind. And if you talk about the flowers should be having only 1 ovule each, because wind is not a sure pollinating agent. So if you are investing into the plant for making multiple ovules, you are wasting your energy. You don't want that to do. You don't want to do that. Basically, the flowers which are there should be having just 1 ovule. So all these important things you should be writing down on the day of exam. Okay, for the flowers that are pollinated by wind. Okay, and also the stamen which is there, it should be long, it should be tall, such that wind can take it. If they are very short, can the wind take it? No. Okay, so what are false fruit? That is also there. False fruit, as in usually our ovary is there which becomes the fruit, but sometimes what happens, the fruits arises from other parts of the plant, other parts of the flower, basically like the thalamus and all. You know the thalamus is where, thalamus looks like this, onto which the flower petal and everything is arranged. So this time the thalamus is also contributing to the formation of fruits, my dear children. That's when you call it false fruit. So in the case of apple, it is formed from the thalamus and also the ovary. Okay, cashew from the pedicel and also the ovary, and strawberry. So all of these are being formed from things apart from the ovary as well. That is thalamus, pedicel. So pedicel is this stalk-like structure which is being formed over here. Got it? So these are things that you should definitely not miss. So insect pollinated flowers are also really important. You should know the characteristics of all these things: wind, water, insect, everything. So wind and insect is the most commonly asked one. You can remember. You can just go through the water one as well. Now describe the structure of dicot embryo with the help of labeled diagram. So over here, dicot embryo you should be knowing. So what is there? Here they have 2 cotyledons, my dear. These are the 2 cotyledons. Above the cotyledons you have the plumule which will be giving rise to the shoot. Okay, and then below it you have the radicle which is covered by the root cap. Radicle gives rise to the root system and everything. Now things above the cotyledons are known as epicotyl, things below it is known as hypocotyl. So hypocotyl has been mentioned over here. Epicotyl is not been mentioned, but you can mention it. So this is just epicotyl which will be basically bulging around and opening into what? Plumule. Works? Cool. Now every time if you can, support your diagram with one-liner thing, any one line, that basically dicot embryo will be having 2 cotyledons to store what? The nutrients up there. That is why it is known as dicots, anything like that. It will be really good if you give an extra line over there. Works? But don't waste your time into it. Now picture related to pollination is given below. Now over here this is the picture given where there is one insect, my dear children, and the flower which is given. You have to say, will this pollination confirm fertilization? Any pollination, my dear children, will it confirm fertilization? No, because pollination is what? Just the transfer of male gamete to the female, reproductive part. Now, who would know that whether this particular male gamete, the pollen grain which is there, it will germinate and transfer down the male gametes or not? We do not know that. So any type of pollination would not confirm fertilization. Please remember that. And now, what are the floral rewards provided by the plants to the insects to revisit? So they have to come back again. So what are the things? Nectar is there. Apart from nectar, pollen grain itself is being provided. So don't think that if plant is giving pollen grains to eat, then how can it pollinate? So some amount of pollen grains would be given to eat, the rest of them will be sticking on their body, my dear children. Anyways, that would be transported easily. Now shelter is also one, a place to lay egg is one big reason, a reward that the flowers give, okay, place to lay egg. Now, mention the characteristics of flower in this pollination other than the floral rewards. So, the flowers which are there, it will be what, my dear children? It would be large, it will be colorful, fragrant and very rich in nectar. If they are small flowers, they would be coming together to form inflorescence. Okay, so that they can be seen very easily. Now, if they are pollinated by flies and beetles, it will be having foul odor. Instead of fragrance, it will be having foul odor, because that is the one that attracts what? Flies and beetles. Cool. So these are the things that you can write, and over here I have written the whole answer. The PDF, you will get it if you join our WhatsApp group. Okay, there the links have been provided, so do not miss it. Yeah, cool. Going ahead, explain the structure of Megasporangium. So over here you have to explain the whole thing, that there is basically what, you can support it with the help of a diagram as well. You have to make the ovary, inside the ovary make the ovule. So in the ovary, what is there? There is placenta, my dear children. That is, so suppose this is the ovary, so there is placenta, there is ovule. The placenta is linked to the ovule with the help of funicle, and the place of attachment is known as hilum. Then the ovule has two integuments, one is this and the other one is this. Now there is an opening over here which you call as micropyle. Okay, opposite side of micropyle is known as chalazal end. The cells which are inside there, it is known as nucellus cells. Okay, so all this thing you should be writing up over here. So ovule is small structure attached to placenta by means of a stalk, that is funicle. The body of the ovule fishes with the funicle in the region that is hilum. Then you talk about integuments. Then you say that there is an opening micropyle. Opposite to it you have chalaza. The integuments enclose a mass of cells with reserved food. What do you call this? Nucellus. Nucellus encloses the embryo sac. So inside it only there will be one cell that will be giving rise to the embryo sac or female gametophyte later on. So that all things you should be mentioning up over here in the structure of Megasporangium. This is a 5-marker thing, so obviously you can explain with the help of a diagram as well, even though the diagram is not being asked. Okay, so first make sure you're writing it and then you can draw something. Now, picture of the mature angiosperm embryo sac is given below. Answer the following that follows. So you should be knowing the ploidy of each cell over here. So this is the embryo sac. There is egg cell, there are 2 synergids, there are 3 antipodal cells, okay, and then there are 2 polar nuclei. So which cell nuclei of the embryo sac produces zygote and the primary endosperm nucleus? Which one is the one that will produce zygote, my dear children? The female egg cell over here. The egg cell and the male gamete will be coming and fusing and giving rise to zygote. What about primary endosperm nucleus? Endosperm nucleus as in the one that is a form of, that is a part of endosperm cell. How do you get endosperm cell? Basically the second male gamete which comes. Okay, there are 2 male gametes. One male gamete fuses with this guy, that is the female gamete. Another male gamete will be coming over here and fusing with the 2 polar nuclei. Okay, or basically the central cell over here which has the 2 polar nuclei, giving rise to what? Primary endosperm nucleus. So you should be knowing that it is a polar nuclei or the central cell which will be giving rise to primary endosperm nucleus. Now what is the ploidy of antipodal cells and the primary endosperm nucleus? That is, it is diploid, haploid, what is it? So let me tell you one thing, in embryo sac all the cells, my dear children, if you see, all of them are haploid in nature, except central cell. In central cell there are 2 nuclei. In central cell there are 2 nuclei. Each of them has haploid content. So this will be diploid. This is diploid because it has 2 nuclei, my dear children. Rest all of them are haploid. So what is the ploidy of antipodal cells? They are haploid. And primary endosperm nucleus, how do you get it? When this fuses with another male gamete. Another male gamete will also have half the number of chromosomes. Yes. So diploid plus haploid is equals to what? Or 2n plus n gives what? 3n, which is triploid, my dear children. So primary endosperm nucleus or primary endosperm cell, if you are asked about the ploidy, it is what? Triploid in nature. Cool. Now, why the endosperm development precedes embryo development? Why do you think endosperm develops first and then comes the embryo? Because the endosperm is the one that will be providing the nourishment to the zygote to form the embryo. So endosperm has to full completely, right? It has to be made completely. That's when it can provide nourishment. That is the reason why it has been preceded. Development is preceded than embryo development. I hope you got the answer. The answers are right over here, my dear children. You have to write it in this format. Please make sure you're writing in points as well. Cool? Going ahead, write any 3 outbreeding devices. Outbreeding devices is you're stopping the plants from inbreeding, from self-pollinating. So what are the devices? So over here you have unisexual flowers. If you have unisexual flowers, obviously the male and the female parts are not together. So there are higher chances that different cross-pollination can also happen there. Now dichogamy is there, where basically the pollen release and the stigma receptivity is not being synchronized. There are 2 types, that is protoandry and protogyny. Protoandry means basically the anther is maturing first than the stigma. Here protogyny, the stigma is maturing first than the anther. So these 2 comes under dichogamy itself. And then there is self-sterility or self-incompatibility. Now this is a genetic mechanism adapted by plants. So even if the pollen grains come by land, the genetic mechanism does not allow the pollen grains to germinate. The pollen grains would not still germinate. That is a genetic mechanism. So these are 3 outbreeding devices that you should be definitely knowing. And in unisexual flowers, my dear children, there are 2 types. You know, monoecious plants and dioecious plants. Monoecious are the ones where there is one plant, and in one plant you have 2 sexes separately. In one plant you can have 2 sexes separately. Okay, so when you have both the sexes on one plant, you will call this monoecious. You will call this condition as monoecious. Now in monoecious also you have 2 conditions. One is like this, and the other one is where in one flower you have both the sexes. Anyway, the condition is you should have both the sexes in one particular plant, right? So here also that condition is suitifying. Here also it is done. So monoecious plants, in monoecious, unisexual are the ones. When they have unisexual flowers, those are the ones that are better for outbreeding. Okay, not like this. This is bisexual monoecious. This is unisexual monoecious. Example of unisexual monoecious is castor and maize. Castor and maize. Now you tell me, will autogamy happen over here? Autogamy means from male to female of the same flower. Will that happen? No. In the same flower there are no male and female. So autogamy is not happening. But what about gytinogamy? Gytinogamy means 2 different flowers of the same plant. Yes, that can happen over here. So autogamy is prevented, but gytinogamy is not prevented. Now if we talk about dioecious plants, dioecious means 2 different plants. In one plant there is only one sex, in another plant there is another sex. So these are dioecious plants. They do not have both the sexes on one plant. So this will assure what? This will assure xenogamy. So there is no gytinogamy, there is nothing autogamy also, there is only xenogamy, and this is the perfect way of outbreeding devices. Example of this is what? Example of this is papaya. Example of this is papaya. So these papaya, castor, maize and everything which is there over here, these are really important for your MCQ questions. Cool? Works? Chalo. Done. So that chapter has been done. So make sure you are going through entire concept that I told you over here, my dear children. And for MCQ question, I'll tell you. Okay, for MCQ question from this particular chapter, make sure you know what is this thing? What do you call that? The flow of microsporogenesis. Okay, that is sporogenous tissue, and from them the microspore mother cell, and then microspore tetrad, all of this thing. Okay, then microspore and pollen grains, this thing. And now about the layers of the microsporangium, that is basically epidermis, then endothecium and things like that. That is very important from this particular chapter. In the case of MCQs, apart from that there are others. So I have included all of them in the 1 marker marathon series. Please go watch it. It is a very important video. Okay, put it in 2x speed and watch it. It will be helpful. Going ahead, human reproduction, which carries 11 or 10 marks. Okay, in different blueprints you have different marks. So first question is, parturition is induced by complex neuroendocrine mechanism. So you have to tell both the things, why it is neuro and why it is endocrine. Neuro as in it is under nervous control. Why it is endocrine? Because it is under hormonal control as well. So what happens? First thing, fetal ejection reflex, you have to mention about that. Where the baby is completely grown, my dear children, and the placenta is also there, what happens? Basically the baby will be trying to move out. Okay, now this will be activating the receptors which are there on the surface, my dear children, and this will give a signal. This will give a signal to the maternal pituitary to release oxytocin. It will be releasing oxytocin. Now what happens? It's not just a baby who will be doing this function. Okay, so baby goes around. Okay, the fetus, which is fully grown fetus which is there, it will obviously push and make the receptors active over there. But one more thing happens, that is in the placenta, the amount of estrogen which is there, it increases, and this estrogen makes the oxytocin receptors really active on the myometrium. So once the baby is moving out, the stretch receptor active, it will send signal to the maternal pituitary to release the oxytocin. The oxytocin will come and land over here and it will create more contraction. So this is both, this is nervous thing where the baby is pushing, trying to push down, receptors getting active and then sending the signal to the maternal pituitary. From here it becomes hormonal, my dear children. It will become what? Hormonal. So that is fetal ejection reflex, the first thing. Okay, the first signal comes from the fetus and the placenta, and that is known as fetal ejection reflex. Post that, okay, when the fetus reaches the uterus, the full term, its head pushes against the cervix, activating the stress receptors. Now nerve signals to the brain. The stressed cervix sends signals to the hypothalamus. Oxytocin release. The posterior pituitary secretes oxytocin in response. Please remember, it is not the pituitary which produces oxytocin. It just stores it. It just stores it. The actual one is hypothalamus, but it stores in the posterior pituitary, and posterior pituitary is the one that releases it. It is not made there. Remember that. Now oxytocin stimulates powerful contraction of the uterus. More contraction lead to what? More positive feedback, and this goes on until the baby is finally moved out. So you have to explain about point to point. First thing, you have to talk about fetal ejection reflex, where you have to mention that it is both the fetus, fully grown fetus, as well as placenta, which is responsible for this particular thing. Cool. Now going ahead with the next question that we have is, in the figure given below, part A and part B, level of hormones which influence the menstrual cycle, study the figure and answer the following. So name the organs and glands which secretes the hormones represented in label part A and B. So over here this is A, this is B, and what do you see? Ma'am, I don't see anything because the brightness is way too much. It's okay. So over here in A there is LH and FSH. Who releases it? It is anterior pituitary gland. It is anterior pituitary which releases FSH and LH, my dear children. Who releases estrogen and progesterone? It is basically the ovary which releases initially. Estrogen and progesterone is being released by ovary. So you write that. Okay, now state the role of hormones secreted from part B on the uterus. So you have to tell what does estrogen and progesterone do. So initially estrogen help in the reformation of the endometrium which is shut down, yeah, and then progesterone also comes along. It will make sure that the whole endometrium is being maintained throughout, okay, and it makes the endometrium secrete certain things like glycogen and stuff like that. So these are the functions of estrogen and progesterone, my dear children. So just a second. Yes, how do you write it? So estrogen, especially during proliferative phase, stimulates endometrial growth and thickening, prepares the uterus for implantation. Progesterone stabilizes and maintains the thickened endometrium, my dear children. It makes it more thick and stabilizes it as well, and it enhances glandular secretion for the embryo nourishment, if at all embryo is being found. Okay, now if fertilization does not occur, both the hormone level drops, leading to what? Menstruation. Again, the detailed version of this particular answer I have already given. Watch the video if you need so. I'm just here to tell you what's important. So this particular cycle itself, whole phases are really important. Just go through it once. Now, during pregnancy, the level of hormones are increased several fold in the maternal blood. Name the hormones and write their importance. First thing, you will be having placenta. Placenta will be releasing certain hormones, right? That is human chorionic gonadotropin, progesterone, estrogen, and human placental lactogen. But apart from that, in your blood, there are certain other hormones also which increases way too much. One is thyroxine. Okay, and apart from that, you have cortisol, and then you have prolactin. These three hormones are also going to increase several folds in the maternal blood, okay, during pregnancy, my dear children. Again, I have mentioned about each of its function. You should be knowing the functions. These are really important. You can get other questions. That is, what is the function of placenta? In the function of placenta, you have to tell it is producing hormones, and the role of hormones is to what? Maintain pregnancy over here. Okay, do this. All these things are really important. Got it? Okay, now let's the functions of human placenta. So placenta is connected to the embryo through umbilical cord. It helps in what? Supply of oxygen and what? Nutrients to the embryo, and also removal of carbon dioxide and excretory waste. You can basically put down into 2 points and then write this point. Okay, what are they giving? What are they taking back? Now it also acts like an endocrine tissue because it releases hormones. I told you the name of the hormones are really important. Can you also just tell what do they do? Little bit. little bit of information. If you think the points are really less, you can jot it down, no problem. Human chorionic gonadotropin basically helps in maintaining pregnancy by maintaining the endometrium, okay, by basically maintaining the corpus luteum. So corpus luteum will keep on releasing progesterone, which will help in the endometrium formation, but there should be someone to maintain the corpus luteum, and that someone is HCG. So all this information you can write it down if you need, so if you think the points are really less, okay. And the placenta is connected with the help of umbilical cord to the embryo. Cool, works. So next thing, briefly explain sex determination in human beings. So over here you have to write that they show XX, XY type of sex determination, where XX is female, XY is what, male. Then you have to draw it and tell, okay, that these are the parents, where XX is the female and XY is the male, okay. Then you have to tell the gametes which are there. So X, it is X and another X, make it in round, X over here and Y over here, and then the progeny which is being formed over here. So you cross this with this, you get XX. That means it is what, a female, right? That it is a female. This and this, you get XY, that is, it's a male. Similarly, you have grown, and then at the end, right, 50% of the chances are there that it could be a male, 50% of the chances there that it could be a female as well. This is the sex determination in the case of human beings, okay. Males over here are heterogametic, are what? Heterogametic. That means they are going to produce two type of gametes, one is X, other type is Y, whereas female is what? Homogametic, as in they are producing same type of gametes, both of them are going to give just XX, okay. So this is the sex determination in human beings. Now going ahead, distinguish between menstrual cycle and the estrous cycle. So menstrual cycle, my dear children, occurs in the case of primates like human beings and monkeys, whereas estrous happens in the non-primates like cows and dogs. So what is the major difference between us and them? Basically our brain capacity, brain volume is much more than them. Okay, now talking about menstruation, here the endometrial lining is the one that shuts down, but in them, if you have seen dogs, they do not have shedding of endometrium, basically it's reabsorbed. Spotting would be there here and there, little amount of blood will be lost, but not too much. Most of the endometrium is being what, reabsorbed. Now sexual activity, females are sexually active throughout the cycle, once they are reproductively active, my dear children, throughout the cycle, they are sexually active also. Now sexual receptivity only occurs during the estrous phase. So there is a phase, that is the estrous phase or heat phase, that's when they are sexually receptive. Now frequency, typically monthly. Every month there is menstrual cycle, my dear children, that is occurring in us, but here it varies. It can be seasonal like dogs and horses, or it can be continuous like cows. Okay, now ovulation timing, mid-cycle around day 14 in humans, or this is during the estrous phase, that's when the ovulation happens. Okay, so if you're not writing this and all, at least if you write this, this and this, it's more than enough. Okay, it's more than enough, because in an NCET it's not given in a very enlarged manner. It's just one line that is given, and out of it they are giving a 3-mark question. So obviously it is not fair to ask, but yes, so you have to be prepared. Got it? Chalo. Going ahead, draw a labeled sectional view of human female reproductive system. Very, very important. So this thing you have to draw, make sure you are making 3 layers of the uterus which are over here, the ovary, and then the parts of the what do you call it, fallopian tube or the oviduct. First thing is the fimbriae, which is from infundibulum. Then you have a broader region, that is ampullae. Then you have a narrow region, that is isthmus. Then you have uterine fundus, this is the uterus. You have the cervix, you have the cervical canal, and then you have the vagina. Cervical canal plus the vagina becomes the birth canal. So all this information are really, very important. Where does the fertilization occurs? Fertilization occurs in the ampulla. Okay, so make sure you're writing it, and make sure you're drawing it really, really well. Next diagram, draw a neatly labeled diagram of sectional view of mammary gland up over here. So in mammary gland, I always tell, if you remember the labelings, okay, if you remember the labelings, you would be able to remember to draw it also. So first thing that you have is lobes, okay, how many lobes? 15 to 20 mammary lobes are there. So if you see the structure, this is one lobe, this is another, this is another. So there are different lobes, okay. In lobes, you have so many alveoli. So the tree-shaped structure is alveoli. The alveoli will be opening into mammary tubules. So lobes, then alveoli, then mammary tubules, my dear children. Each tubule will be opening up into mammary duct. The duct which are there over here, they will be opening into ampulla, which are broader, and ampulla will be opening into lactiferous duct. Over here you have the nipple, over here you have areola. It is surrounded by fat tissue, my dear children. At the back of it you have ribs, in between that you have muscles. Okay, so picturize the diagram, you would be able to remember it better. Okay, now draw a neatly labeled diagrammatic sectional view of male reproductive system. So I saw a comment, ma'am, is this diagram important or shall we draw the other diagram? So if it comes like draw the diagrammatic view of male reproductive system, then you can draw the other diagram. Otherwise, if it is sectional, if sectional word is mentioned over here, my dear children, that means you have to draw this particular thing. Okay, in that case obviously the testis is there. Okay, from here, from the testis, sperms are being transported with the help of vas deferens, okay, towards the urethra, which comes from the urinary bladder, but it does not directly go and open there. Its seminal vesicle will be there, it will be opening together in the form of ejaculatory duct directly into the urethra right here, and then from here it will go to the genital organ, external genital organ, that is the penis. Over here the enlarged portion is glans penis, it is covered by skin that is known as foreskin, my dear children. And the talking about the accessory gland, one is seminal vesicle mentioned over here, one is what, prostate gland, which is like a donut, through which basically through this only the urethra will be passing down. And then you have two bulb shaped thing, that is bulbourethral gland. Just before the opening into the genital organ, the bulbourethral gland will be giving out its secretion. So these are the things that you should definitely do. Backside of it you have the rectum and the anus, and that is the spine. Okay, and the urinary bladder is connected to the via the ureter to the kidneys. Okay, so this is a sectional view of male reproductive system. Now write the schematic representation of spermatogenesis. Very important question. You might be asked to differentiate between spermatogenesis and oogenesis right there. So you should be knowing about thing. In spermatogenesis, from one spermatogonia, basically one primary spermatocyte gives 4 sperms, whereas in the case of females, one primary oocyte will be giving you 1 ovum only. So that question has been done. I have asked also. So there are basically 500 spermatogonia. How many sperms will be produced? So 500 into 4, you do like that. So it is 2000 sperms will be produced. What about if there are 400 oogonia, that means 400 ova will be produced. You don't do it into 4, because one oogonia will be differentiated into one primary oocyte, that will be differentiated into one ova at the end. Not differentiating, it will be dividing and forming one ova at the end. So over here, know that spermatogonia is there, it will divide itself into way too many number. It will undergo differentiation, it will form the primary oocyte, primary spermatocyte, which is there over here. This primary spermatocyte, it is diploid. Remember, all of them are diploid until here. When you start meiosis, after first phase of meiosis, it will become half. So it was diploid over here, 2n number of chromosome was there, but now it has become haploid. So secondary spermatocyte will be having half the number of chromosome. Now again, this will undergo meiosis number 2, that means it will just divide, it is still haploid. From here everything is haploid. So you get spermatids, my dear children, these are spermatids. So they are like kids, they are immature, and then they become mature into what? Spermatozoa. So this is spermatozoa. What do you call this process as? This is known as spermiogenesis. Spermatids to spermatozoa differentiation is known as spermiogenesis. And the removal of the sperms, or basically the transport of the sperm outside the seminiferous tubules, is known as spermiation. So you should be knowing about all these 3 things: spermatogenesis, spermiogenesis and spermiation. Again, an MCQ question, and also the flow of this is also an MCQ question. Now write the 2 events that occur in the ovary and uterus during follicular phase of menstrual cycle. I told you the phases are really important. Follicular phase is also known as proliferative phase. Then there is luteal phase, which is also known as secretory phase. And then in between follicular and luteal phase, there is ovulatory phase, where the ovulation happens. Before all this comes menstrual phase, where the blood is being shut down or the endometrium is being shut down. So all these phases are really, really important, and you should know what is happening in all these phases. Okay, so you are asked about follicular phase. In follicular phase, the follicles are developing in the ovary, my dear children. The primary follicles are maturing into graphene follicle. Talking about uterus, in the uterus what is happening? The endometrium, which is shut down, it is growing back with the help of estrogen. So the endometrium is regenerated through proliferation, which is why it is known as proliferative phase as well. Cool. It's a very important question, 5 marker. Now going ahead, write the role of the following: Sertoli cells, Leydig cells, oxytocin, menarche and menopause. So my dear children, Sertoli cells, what are Sertoli cells? They help in providing, they are nerve cells, they provide nourishment to spermatogonia. Whereas Sertoli cells are present inside the seminiferous tubules. Inside the seminiferous tubules, there are 2 cells: spermatogonia, which are going to form the sperm, and Sertoli cells, which are going to provide nourishment to these growing sperm cells. Okay. Now Leydig cells are just outside these seminiferous tubules. So between the seminiferous tubules, there is some space, interstitial space. In that you have the Leydig cells, which will be producing what? It will be producing, Leydig cell produces what? Hormones, the male sex hormone, that is androgens. So you have to talk about that over there. Then oxytocin and parturition, my dear children. Oxytocin basically help in contraction and relaxation of the myometrium, which will obviously help the baby move out of the uterus. Now menarche is the first time women get her menstruation. So it's the first day of or basically the first time, what do you call it, first menstrual period, signaling the start of the reproductive ability. Menopause is what, the end of the menstruation, marking the cessation of fertility. This term is really important. Cessation of fertility as in now they are not fertile anymore. Okay, they would not be having baby anymore. So these are the things that you should be writing, and with that we complete that chapter as well. Now reproductive health is there. This is a very small chapter, gives you a lot of marks, 6 marks. Okay, so that 6 marks you have to get it. There's no chance you're going to miss it. So mention the 2 medical grounds on which pregnancy are subject to termination. Okay, so when it is risk to the mother's health and when there is a risk of fetal abnormality as well, that's when medical termination of pregnancy is being approved by what in the Medical Termination of Pregnancy Act, my dear children. Okay, risk to mother's health, and also when the sex is not contiguous, as in rape and all, where there is no permission being asked, at that point also it can be done. So if continuing the pregnancy poses a risk to physical or mental health of the mother, that's when it can be done, or risk of the fetal abnormality, either way. Now, what are emergency contraceptives? Write their hormonal combination. Emergency contraceptive as in the one which can be taken within 72 hours of coitus, unprotected coitus, and still, okay, still you do not want pregnancy to happen or anything to happen, that's when you have it. So emergency contraceptive would be rich in 2 hormones, progesterone and estrogen. Okay, progesterone or estrogen, either way you can write it, a combination of both or either progesterone. Why? Because usually when they are in usual level in our body, they will help in ovulation, they will help in implantation, and they will do all the things. But when they are in abnormal level inside our body, they are in too much level in our body, they are going to do the opposite. They do not allow ovulation to happen, they do not allow implantation to happen. So instead of maintaining the endometrium, they will have a thin endometrium. Then how will implantation happen? How will the embryo will come and what's the implant over there? Get it? So that is being prevented. Apart from that, yeah, so those are the 2 things that you can possibly write. Okay, and so are the pills used to prevent pregnancy after unprotected sexual intercourse or contraceptive failure as well? Now they are intended for use within a short window, that is usually within 72 hours after the event. Administration of progestrogens or progesterone estrogen combination or IUD is emergency contraceptive, my dear children. Cool. Now what is infertility? Give reasons for infertility. Infertility is basically inability to produce what? Offsprings in spite of unprotected sexual intercourse. Okay, now reasons can be many. It can be physical, congenital, that is genetic, diseases, drugs, immunological or psychological as well. Or also you can talk about if the sperm count is less, if the ovum count is less, if the uterus is not allowing implantation to happen. These can also be reason for infertility to be caused. So those comes under physical things, but yes. Okay. Now, what are the features of ideal contraceptive? Mention the natural methods of contraception. Again, all the methods of contraception are really important. Okay, so over here, first thing, features of an ideal contraceptive. It should be user friendly. Yes, it should be user friendly. It should be easily available. It should be effective and reversible with no or less side effects. Should not interfere with the sexual desire of the person as well. Okay, it can be used, should be used by everyone. It should be easily available. It's effective and reversible. It should not have permanent effect and should not interfere with the sexual desire. These are the 4 features of ideal contraceptive. Now mention the natural method of contraception. In that there is periodic abstinence where they do not have sexual intercourse during the fertile window. Withdrawal method where before ejaculation only the penis has been withdrawn from the female body. Now lactational amenorrhea is when lactating mother would not be ovulating. Why? Because of the hormonal placement changes. Prolactin will be way too much, which does not allow ovulation to happen. So this time menstruation will also not be happening. Hence that particular person will be infertile for a certain period of time until she is lactating profoundly. Okay, so these are the 3 natural methods of contraception. Now, the popular and effective contraceptive includes IUD. Mention the types of IUD. Again, I told you, all the contraceptive methods are really important. So, in IUD, you have non-medicated IUD, that is lipase loop, which are not going to produce any hormones in our body. It is just going to act like a foreign thing. If there is something foreign in our body, obviously our body will react to it. It makes the uterus unsuitable for implantation. Now, copper-releasing IUD is there. Example, you should know that is copper T, and you have copper 7, and you have multiload 375, my dear children. All of them are going to release copper. And what does the copper ion do? Copper ion does not allow the motility of sperms. Sperms would not be able to move around. It will be really toxic. Now, sperm are not able to move around. Will they go and fertilize the ovum? No. So that is one thing. Now you have hormone releasing IUD, my dear children. In hormone releasing IUD, you have LNG20, you have progesterone. All of them, they are going to release hormones, that is progesterone or levonorgestrel. Now over here, my dear children, what does this do? I already told you. Ovulation has not been allowed. The thin endometrium is there, not thick. Thin endometrium is there. And one more thing happens. You know the uterus. Okay, in the starting part of the uterus you have the cervix. So in the cervix, if there is a plug being formed, a mucus plug being formed, can the sperm enter? No, the sperm would not be enter. So this mucus plug is also formed because of the same reason. Okay, so these are the things that you should be definitely knowing about IUD. Please remember the examples, very important. It can come in the form of MCQs as well. Okay, now they can ask you this and this comes in what? So you should know, no. Now, suggest 3 simple principles through which we can prevent sexually transmitted diseases. First thing, practice safe sex, use condoms during intercourse. Second thing, have a monogamous relationship, because if there are more number of people, there are high chances of getting what? STDs. Now, avoid sharing needles. That is not a direct point. Why do I say so? Because sexually transmitted diseases are believed to be transmitted sexually through the sexual act. Now over here, these diseases are also the ones that are spread through body fluids like blood. So when you are using needles and things like that, you are having higher chances of getting these diseases like HIV and hepatitis, which are STDs and which are spread through blood as well. You get it? So you have to mention that it is said that they are transferred through what? Body fluids, which is why blood can also be a reason, which is why avoid sharing needles can also be a point. Start with that and then you can write this. Okay? So these are the 3 things. In your book, it is written that you should be talking to the counselor. So that is obviously not a way to prevent it, but yeah. Okay, so what is contraceptive? Briefly explain 2 barrier methods and 2 surgical methods. So my dear children, in contraceptive, what is contraceptive? Basically, it is used to control the childbirth and unwanted pregnancy. That is contraceptive. Now, if you talked about barrier method, again the answer is over here is natural method. I am talking about barrier method. In barrier method, you have condom. Okay, you have condom, which is going to prevent the sperm and the egg to meet. Okay, physically, it is acting as a physical barrier. Apart from it, there is female condom as well as diaphragm as well as walls. Okay, there are walls, there were cervical cups. So basically, this is used to cover the cervix region. Again, it is acting as a physical barrier. But when we are using vault and all, you're making sure that you are putting spermicidal things as well. As in the thing that could kill the sperms as well. If at all the sperms are moving around, they can be killed. That's why. Okay, so these are the 2 barrier method which is there. Now talking about the surgical method, you have vasectomy and tubectomy, my dear children. In vasectomy, you are going to cut the vas deferens. In tubectomy, you are going to cut the fallopian tube. Okay, you're going to cut the fallopian tube. So fallopian tube is where the ovum is transported. So if you're cutting that, the ovum would not be transported. In vasectomy, you're cutting the vas deferens which transports the sperm. Again, the sperm would not be transported. You got it? So one thing that you should be knowing, in both the cases, still ovum is produced, still spermatogenesis takes place. Remember this, still spermatogenesis takes place, still ovum is produced, but it is not just transported. They will be degenerated, but they are produced, please remember it. And in vasectomy, one more important question, in the semen that is being produced. So male, they will still produce semen. In the semen, but there is only seminal plasma, as in the secretion of the ascitic glands. There is no sperm. There is no sperm. So there is no sperm found in the semen, please remember it. Okay? So the question can come, in vasectomy, sperms are not produced in the epididymis. Array, in epididymis still everything will be happening. It's the vast difference where the sperm are not transported because it is being cut and tied. So don't do that blunder. Okay, everything will be happening normally, just that it is not transported. That's all. Okay. Now principles of inheritance and variation, that carries 13 marks. So just want you to take a 1-minute break and come fast, drink some water. I will also come drink water and be coming in a second. Okay. Okay, I am back with the business. Okay, so business, oh my god, what happened? Yeah, principles of inheritance and variation, which carries 13 marks. The question goes as follows. Now it will contain the most important questions, okay, because obviously 13 marks is not a small deal, right? So write a short note on sex determination in birds. So you should be knowing, in birds it is exclusively going to show female heterogamety, but the females are going to produce 2 types of gametes, unlike in the case of human beings. So here you follow ZZ and ZW type of things. ZZ and ZW type of sex determination, where ZW is for female and ZZ is for male. So this is the only place where you see female heterogamety. You should go see the answers. Say the females will be producing 2 type of gametes, and if the gamete carrying Z is fusing with another gamete carrying Z, then it will be a male. If the gamete carrying W will be fusing with another one that is carrying Z, it will be a female. All this information has to be provided one by one in points. Okay, model question, paper question, you should not miss it. Going ahead, what is haplodiploid sex determination? In this, the example is honeybee, my dear children. You have to tell the organism. It is honeybee that shows haplodiploid sex determination. What happens over here? In the females, it will be diploid. The males will be always haploid. Okay, so here the female has 32 number of chromosomes, the male has 16. It will undergo meiosis. At the end, it will produce gametes which is having 16 number of chromosomes. Half will happen in meiosis, right? Over here, in the case of males, they will just undergo mitosis. They will increase the number, and there are 16 number of chromosome in there. Now suppose this gamete fused with this gamete, it will be 32. It will be 32, then it will be a female because it is diploid. It is diploid. But over here, if the 16 is not fusing with anything, any gamete, it will transform into a male, and this process is known as parthenogenesis. Do not confuse with parthenocarpy. Okay, it is parthenogenesis, where the gamete which is not fertilized, it will just transform into what? Males. So you just have to write about all this thing. Haplodiploid, where do you find it? And males develop from unfertilized eggs and are haploid. They develop from fertilized eggs and are diploid. A queen bee lays eggs. If an egg remains unfertilized, it develops into male drone. If an egg is fertilized, it develops into a female worker or a queen depending on environmental and dietary factors. So if there is no queen up there, this particular queen is old again and about to die, basically obviously at this point of time another queen is being produced. Otherwise, so how they are being treated with royal jelly and things like that, that's when you get a queen. Okay, otherwise it will be just a worker who will be working around. So that's a sad thing, but yeah. Now, write the genotype of the parents when their children are A, B, O blood group. So, these are the things. So, how do you show it? So, if you basically have an idea, and if there all the blood groups are given over here, that means the parent will be what? IA, IB, the other parent. Is it like that? Can it be like this? Can it be like this? Another parent can be IB. In that case, my dear children, there is no O blood group that is going to come. So if there is O blood group mentioned over here, that means both the parents will have at least one allele which is O. Because it can be expressed only in recessive condition. And if B and A both are mentioned, that means one allele will be A and the other one will be B. This will be the condition. This is the parent's genotype. So you should be knowing the genotype of the parents would be like this. So you can mention it like this, or you can say it can be IA and I. Okay, this also means the same thing. Cool. Going ahead, write 2 symptoms of Down syndrome and write the chromosome number in Down syndrome, my dear children. So 2 symptoms of Down syndrome is basically individual is short-statured with small round head, furrowed tongue is there, my dear children. It's partially open mouth. The palm is broad with very characteristic palm crease. There is physical, psychomotor and mental development which is retarded. And if you're talking about the chromosome number in Down syndrome, what happens? Usually 21st chromosome we get a pair, right? One from a father, one from a mother. But over here there is one extra chromosome which makes it trisomy of 21st chromosome. And 21st chromosome, that means you're talking about autosomes. They're not sex chromosome, they're autosomes. So initially we had 44 autosomes, and it can be XX or XY condition. If this is the condition, it will be X. If this is XX, it is a female, and if it is XY, it is a male, right? But in the case of Down syndrome, there is one extra autosome, my dear children. So it will be 45A plus XX, that is a female who has Down syndrome, or it can be 45A plus XY, that is a male that has Down syndrome. So you have to explain like that. So do not say 47, instead you can just write it like this. That will be more than enough. Say this is a male, this will be a female. Cool. Now, define point mutation, give an example for point mutation. What do you mean by point mutation? When single base pair is being replaced, my dear children, it is known as point mutation. Example, you have sickle cell anemia. You know, in sickle cell anemia, what do you have initially? GAG, but it is replaced by GUG. A is replaced by U. Always remember A comes before U. So A is replaced by U. Okay, GAG coded for glutamic acid, but GUG codes for valine. Okay, that is another amino acid. So this changed the whole structure of hemoglobin molecule. So you can explain that. That is an example of point mutation. Okay, change in single base pair of DNA is known as point mutation. Classical example is this, and that's all. Cool. going ahead. What are the conclusions drawn by T.H. Morgan from crossing experiments in Drosophila with respect to linkage? What are the conclusions drawn by T.H. Morgan from crossing experiments in Drosophila? So over here, always remember, if the recombination is more, that means you're talking about suppose this is a gene and this is a gene, and over here this was the initial condition. After some point of time, what happened? I'll tell you. When the things were exchanged, right? When the things were exchanged, this thing, this gene went up here, and along with it, this also went. This part came over here, this part came over there, went over there, my dear children. Okay, but the problem is these genes are still together. That means there is no much amount of recombination that you can see. So that means when the genes are closely linked to each other, there is less recombination. Remember, closely, I have written over here, let's see that. So the higher the linkage, that means when the genes are closely linked to each other, the lesser is the recombination. Obviously, because they will be always together, and if they are far from each other, then obviously they can move around independently. So if they were far from each other, my dear children, the same scenario, the situation would have been like this. The situation would have been like this. So here both the chromosomes are there, so both the genes are there on the same chromosome, but here they are on different chromosomes. So again, more recombination is there. Why? Because they were far away. So in other words, the genes which are tightly linked will show lesser recombination. The genes which are loosely linked, they show higher recombination. This was the conclusion drawn by T.H. Morgan on his experiment of linkage. Fine? Cool. Now, what is test cross? Write its significance. So how do you write the significance of test cross? So over here, for example, I have a tall plant. I do not know whether it is capital T tall or it is capital T small t tall. That is, it is homozygous or heterozygous. That is when I do test cross. Basically in test cross, what do? I make it cross with a recessive parent plant. So over here, basically there is yellow. Okay, there is yellow color seed which is being produced, but I am not sure whether it is capital Y or small y. So what do? Both the things that I have over here. For example, I just take the plant and try to fuse it with the small y. If the condition was like this, what will happen? All of them are going to produce capital Y, small y. All the things are going to produce capital Y, small y. That means all of them will still be yellow. But if the parent was like this, then what will happen? This and this crosses, you get capital Y, small y, okay, it will be yellow. This and this crosses, it will be capital Y, small y, that means this is also yellow. But what about this and this? This is small y, my dear children. So you get ratio. 2 of them will be yellow and 2 of them would not be yellow, it will be green in color. That means the parent was like this. It is heterozygous, not homozygous. You get it? So that is the use of test cross. It is a kind of back cross where F1 plant is crossed with its recessive parent. It helps in finding the F1 plant is homozygous or heterozygous. So it is, if it is homozygous, I can take this pure breeding plant and try to fuse it, because if I want yellow seeded plants, then I would be basically crossing this plant only. I don't want this, because this can show other character when I keep on crossing, but this won't. This is pure breed. Okay, so that is the thing. Now, going ahead, what is phenylketonuria? Write the cause and symptoms of phenylketonuria. So phenylketonuria, I have explained. You should be telling that it is autosomal linked recessive disorder. That is the first line that you should be having. Second thing, what happens in this disease? The gene which codes for phenylalanine hydroxylase is being mutated over here, which is why this enzyme is not produced. What does this enzyme do? That is the next point. This enzyme helps in conversion of phenylalanine to tyrosine. Now, what happens when it is not converted? When it is not converted, phenylalanine will accumulate in the form of phenylpyruvic acid, and this will cause mental retardation. That is the symptom. So symptoms you can write separately. Accumulation of this in the brain results in mental retardation, and also phenylpyruvic acid and other derivatives are also excreted through urine because of its pure absorption by kidney. Now infants have lighter skin, hair and eyes than siblings. Why, my dear children? Why? Because phenylalanine should be converted to tyrosine. Now tyrosine is one very important thing that is responsible for the formation of melanin, that is responsible for pigmentation of our skin and things like that. So basically, when this melanin is not being produced, do you think the pigmentation would be really good? No, it will be hypopigmentation. The hair will have less pigmentation, the skin will have less pigmentation and things like that. So that is a secondary thing. That is a secondary thing. So with this you get two understandings. That is, it is an example of pleiotropy. One particular gene is somehow is responsible for two characters. One is production of tyrosine, another one is production of basically what? Production of reduced melanin. That is also one thing, which is why hypopigmentation and everything is happening. Get it? So these are things that you should definitely write for phenylketonuria. Now going ahead, T.H. Morgan selected fruit fly for his genetical experiments. Why? Give 3 reasons. So you have to tell the reasons one by one. They could be grown very easily. They have a very short lifespan of up to, let's say, 2 weeks. Single mating could produce so many offsprings. There was a clear differentiation between male and the female sex and the things like that. Now there is one more question. What is it? What are chromosomal disorders? Chromosomal disorders as in when there is a problem in the structure or the number of chromosomes, and which is why you get some diseases. That is chromosomal disorders. The disorder in human are caused due to absence or excess or abnormal arrangement of one or more chromosomes are known as chromosomal disorder. Example, Down syndrome, where you have one extra 21st chromosome. Turner syndrome, where you do not have, where you just have one X chromosome, there is no other chromosome. There is no sex chromosome. There is only one X chromosome. Okay, there is no other sex chromosome. That is what I meant. Klinefelter syndrome, where you have one more extra X chromosome. Initially it was X and Y, but you have one more. So you get it? That is extra chromosome that is being done. So they all are what? Chromosomal disorders. Now, few autosome linked recessive gene blood diseases occur in human population. Among them, some are related to qualitative and quantitative problem of synthesizing blood proteins. Example, explain. So basically, my dear children, you have to understand one very important thing. Autosome linked recessive gene blood disorder is asked. So one question, one child asked me in the route session, that is, ma'am, why hemophilia has not been considered? Bacha, hemophilia is not autosome linked. It is sex linked disorder, gene disorder. It is a blood disorder, I agree, but it is sex linked. So the chromosome over there, the sex chromosome over there, that is affected. The gene over there in the sex chromosome, that has been affected. But over here, genes on the autosomes are being affected, which is why you have these autosome linked recessive disorders. You have to give 2 examples. One should be qualitative, one should be another, one should be quantitative. So qualitative one is sickle cell anemia, my dear children, where what is happening, where because of mutation in the HBB gene on chromosome number 11, okay, that is the one that codes for beta globin chain. So this particular gene is affected over here, which is why you get a defective beta globin. You get a beta globin, but that is a defective beta globin, and because of this defective beta globin, the one that carries it, the cell, will also become sickle shaped. So this is a qualitative disorder. The quality of the cell is being dropped. The quality of the protein which is being there, it is being dropped. It is caused why? Because there is mutation. Okay, GAG is being converted to GUG. Instead of glutamic acid, you have valine, and this whole thing changes the whole structure of beta globin chain of hemoglobin. Again, I am telling you, it is still produced, but it is defective globin chain which is being produced, which is why it is qualitative disorder. Talking about thalassemia, it is quantitative. Why? Because it can happen in 2 things. Okay. One is alpha globin chain and the beta globin chain. You know, hemoglobin has 2 chains, alpha and beta. In sickle cell anemia, you are talking about beta globin chain. In thalassemia, you are talking about both alpha chain and beta chain. Now, for alpha chain, for production of alpha globin, you have 2 genes, HbA1, HbA2. Both of them have their own alleles. 2 alleles here, 2 alleles over here. So, 2 genes on the same chromosome, that is chromosome number 16. Okay. Now, for HbB, you know it. Where are the genes? It is on chromosome number 11. Right. Now, the thing is, here mutation can be happening in one allele. For example, I take this particular chromosome. So, this is HbA, 2 alleles of HbA, and these are 2 alleles of HbA2. Now, suppose this and this is mutated, still others are producing. But the amount of the production alpha globin chain will be less. If 4 of them are affected, that means it is not produced at all. So this is a quantitative disorder. The quantity is being reduced. You get it? Similarly, in the case of beta globin chain, now both the alleles over here, just a second, sorry. Okay. Both the alleles over here, if they are affected, again it is not being produced at all. If one allele is affected, it is produced, but very little amount of it is produced. You get it? That is why it is a quantitative disorder, my dear children. Okay, so you have to explain that, and always remember, hemophilia is sex linked, not autosomal disorder. Okay, you are going to make a mistake if you do so. And in hemophilia, what happens is the clotting factor is not being produced. There is a protein, and a cascade of proteins are there, in which one particular protein is not being produced because of mutation, which is why clotting does not happen, and that person will keep on bleeding. Get it? So I'll just stop this particular video and I'll just save this and I'll come back. Okay? We have give reasons for the following. A simple cut result in non-stop bleeding in hemophilia. As I told, what is happening? Basically, the clotting factor gene that is being affected, which is why there is bleeding. Turner syndrome affected females are usually sterile. Why? Again, because in Turner syndrome there is only one X chromosome. That is why there will be rudimentary ovary. It is underdeveloped ovary. It won't be producing what? Ovum. So that's why it will be sterile. In Morgan's dihybrid cross experiment in Drosophila shows that flies having genes for yellow body and white eyes exhibited less recombination. If it is less recombination, that means the genes are closely linked to each other. That is why they are less, there is less recombination. They are closely linked, tightly linked. Okay. Now, inheritance of skin color in humans show different phenotypes. Why? Because inheritance in skin color is an example of polygenic trait. That means different genes are there coding for one particular character. It is polygenic. Usually where there is one gene coding for one character, but over here it is polygenic. Okay. So different alleles being found. Depending on that, you will be finding different phenotypes. Cool. Now, accumulation of phenylalanine in the body of phenylketonuria affected individuals. Why does it happen? As I told, there is no production of enzyme, that is phenylalanine hydroxylase. That gene is affected over here. Just to explain you this particular thing, that is skin color, otherwise you might not understand. So, listen. So, suppose I have genes over here, 3 genes. A, this is allele for both genes. Okay, this is B gene, this is C1. So, over here, this will lead to what? The darkest skin tone. Okay, this darkest skin tone AA, BB, CC. Suppose here the allele is changed to small a, that means the skin color will be a bit lighter. Suppose both of them, oh my god, okay, both of them are AA, it will be more lighter. If both of them are BB, more lighter, both of them are CC, the lightest skin tone will be obtained from here. You get it? So here, depending on what alleles we have different skin color. That is why we have different phenotypes being observed. So you have to explain like this. I have explained everything. You can just have a quick read of it. Okay. Now going ahead, define aneuploidy, give 2 examples of aneuploidy. Aneuploidy is when there is no proper non-distinction of the chromosome, when they are not properly segregated. That's when you get extra chromosome or you might lose some chromosome. Example is Down syndrome, Turner syndrome and everything. Now mention the karyotype of Klinefelter syndrome of an affected individual. In Klinefelter's, I told you, usually we have 44 autosomes and then we have XX or 44 autosomes or XY condition. In Klinefelter's, what is happening? The autosome number is not being hampered anyway. There is extra XXY. So over here, can I just say 45, 46, 40, 45, sorry, 44, 45, 46, 47 chromosomes are there. So I'll write this condition as 47XXY. I can write like this or I can write like this. Either way, I'm going to get marks. Cool. Now write the symptom. What is the symptom? They will be having masculine nature, they will be tall statured, but they will be having gynecomastia, that is development of breast, which is found in females, not very common in males, right? So these are the things. Infertility due to lack of spermatogenesis and everything is also very important. Now this line you don't have to mention, rest everything you can put down. Cool. Going ahead with the next thing, what is incomplete dominance? Explain its effect in reference to flower color in snapdragon. Over here you have to explain the snapdragons, that is Antirrhinum majus example, my dear children, where the red and the white flowers are being crossed and you find pink colored flowers and then you cross it back again. Then you find 1 red, 2 pink and 1 white color flowers, that is 1:2:1 ratio. Genotypic and phenotypic ratio is the same. Why is it known as incomplete dominance? Because the capital R is not completely dominant on the small r, which is why there is pink color formation, my dear children. So you have to explain this in the format of what's a schematic representation, and also you can write it down like this. Just go through your answer really well, write it in points, make sure you're writing in points. If you're not writing in points, you're going to lose a lot of your marks, and make sure it's not clumsy and things like that. Write in proper way. Okay, wherever you need to underline, it is 1:2:1, underline that. Okay, so that you're showcasing your answers. You get it? All the flowers, F1 generation flowers are pink in color. It's okay, underline even if you have written. Got it? Now write the schematic representation of one gene inheritance Pisum sativum, that is garden pea. One gene inheritance, as in you're talking about one character only. So over here you can talk about the tall plant being crossed with the short plant. Okay, capital with short small dwarf plants. Make sure you're writing the parents, you're writing the gametes, you're writing the progeny, that is F1 generation properly, and then you're saying that it is capital T small t, this is dominant over this, which is why all of them are tall. Okay, now what do? You do selfing. In selfing, again you do the same thing again, and then put down the genotypic and phenotypic ratio properly. If you're not writing the genotypic and phenotypic ratio, you're going to lose your marks. Okay, so please remember to write it. Okay. Just a second, okay. there is some problem up over here. So with that, with this particular question, we are ending what? We are ending our principles of inheritance and variation. Now we will get ahead with molecular basis of inheritance, but these questions kind of jumped in between. Just a second. So over here we have molecular basis of inheritance, that is the next chapter. But before getting into the next chapter, I want to talk about something really important for you guys. So you know there is KCET exam coming up, do you know that? So KCET exam is for getting into the colleges of Karnataka. So if you are from Karnataka, if you have studied in Karnataka for so many years, you are literally very much eligible for this, and I think you should not be missing out this opportunity of giving this exam. Most of you might already know about it because you might have been preparing for this since long. So my dear children, what is the help that we are providing to you? So at the end point, obviously there is only one, let's say, very less amount of time left, which is why we thought we should be putting down some test series or a crash course for you. So you have two options. If you're someone in Bangalore, you can come to our campus. You can learn with the best teachers over here, my dear children, because we are introducing Lakshya KCET crash course over here, where there will be one-on-one doubt session, guided study hours, everything available to you. This is a 21-day crash course. You have to be here. Okay, you can be day scholar, or this is a residential program also. If you want to stay and learn, you can do so. Okay, now if you're someone who feels that I cannot come to Bangalore, ma'am, is there any online thing available? So you have Abhyas KCET test series as well, where there will be 35 full syllabus and chapter wise test available to you. There will be detailed video solution for each of the questions which are being put up there, and chapter wise one shot revision videos as well. Okay, and it is done by analyzing the past 5-year question papers, all for ₹1499. So if you are someone who is in PU2, obviously you are in PU2, so you can take this because you will be getting the PU2 test series also for ₹1. Okay, so it is actually 1498. So you add ₹1 and then you get this also. If you don't want that, don't go for it. Okay, so it's about KCET. So if you're someone who is interested into getting colleges in Karnataka, then you should be looking forward for it. This is a genuine thing. Okay, nothing very promotional. Obviously we have to tell you, otherwise you would not know it. So we have to tell you in all the videos, and we would be telling it because effort is being put over here. So it's not something that you're getting it for free. Obviously you are getting it for money. We are also doing it because there is a purpose to it. You get it? So if at all you're interested, you can go ahead with this. The link is provided in the description box below. You can go ahead and check with it. Now the good thing about Karnataka colleges is that it gives good placement. So with that, if you're someone for going for engineering background, that will be much better for you. But for other courses also, you can just have a quick revision. Okay, just go through it and see how does it work, and then you can go ahead with this. Yeah, chalo. Now, molecular basis of inheritance, let's start the first question. Which sequences of bases transcribed from the DNA are found both in hnRNA and mRNA? Now, hnRNA is the thing that you get from the DNA. When you do transcription, the first thing that you get from the DNA is hnRNA. And in hnRNA, it will be having introns. What are introns? The non-coding regions. It does not code for any genes or any protein, anything like that. So, there are exons and there are introns in the DNA, and that is exactly copied in the hnRNA. But in the hnRNA, what do? You do processing. You cut down the introns and then you join the exons together. So what is common over here? This thing that is being found is this mature mRNA. Now what is common between this and this? Both of them will be having exons. So the sequence which would be there in both would be exons. You have to write that this. The sequences or bases transcribed from DNA that are found both in hnRNA and mRNA are exons. Exons are what? They are coding regions. Okay? Now, mention the levels where gene expression can be regulated in eukaryotes, and how many levels can it be regulated? Transcription level, I may or may not allow transcription to happen. Second thing, processing level, I may or may not allow hnRNA to mRNA formation. Third thing, transport of mRNA. This mRNA has to go out for translation, right? So transport of mRNA can be stopped if I do not want to. Okay. Now translation level, if I do not want translation to happen, I can stop that as well. So there are 4 levels. There are 4 levels where the gene expression can be regulated in the case of eukaryotes. So those are these 4 things. What about prokaryotes? If you know the answer for prokaryotes, tell me in the comment section below. Okay, how can they be regulated? Now write a note on the role of sigma factor and rho factor in transcription in prokaryotes. So what happens in the case of transcription in prokaryotes, my dear children? First thing, the RNA polymerase, when it comes to transcribe, says that I need sigma factor. Without sigma factor, I am not going to initiate the process of transcription. So for transcription to happen, the initiation to happen, you need sigma factor. So it binds to the RNA polymerase, you write like that. Rho factor is basically needed during the end of transcription. So rho factor will come and attach. That's when the new strand of mRNA that is being formed is separated, and also everything just moves away from the DNA strand. Okay, that is the use of Rho factor. It helps in facilitating the termination of the process of transcription. Works? Now, what are VNTRs? VNTRs, variable number of tandem repeats. So, in each of our body, we have different VNTRs, my dear children. I might be having GCCA, this might be repeated like 10,000 times in my body, my dear children. In your body, there could be other sequences, and now they are very unique to you and me. Very unique to me and you. So, I have a blood sample over there, and in that I find the code which is being repeated. I make a complementary code for it, and I take my DNA. Basically, that particular complementary code will be binding to my DNA because it is the exact thing that I have over here. So, how it is very useful in identifying criminals, my dear children? First thing, I will be isolating the DNA, I will be cutting down the DNA, I will be separating them, the DNA fragments, my DNA fragments. Okay, what will happen? They all will separate, and then I am going to take it on the nitrocellulose sheet. Now, I have it on the nitrocellulose sheet. Now, what will I do? The blood sample which was there, from there also I got the DNA. Some DNA was there, I made a complementary strand of it. Now I have the complementary strand of it. This complementary strand, if I run through this, it will obviously go and bind to mine because it was mine. I have the same sequence, you get it? This is how I find the criminals. So this thing you should be knowing. So hybridization using labeled VNTR probe. This is a probe, and that is labeled by radioactive things. Now it can be determined by using autoradiography and things like that. Okay, so that is a whole concept. So you should be writing point to point properly. Now, write the differences between euchromatin and heterochromatin. Euchromatin is basically what? When it is not very much coiled. The chromatin is not very much coiled, it is euchromatin. Over here, since they have little spaces, transcription can still, this is DNA, so transcription can still happen over here. So this stains very light and transcription happens over here, so they are transcriptionally active. Heterochromatin is when it is very much coiled, it is transcriptionally inactive, my dear children, and stains very dark. Okay, now name the virus used in Hershey and Chase experiment. The virus was bacteriophage, which was going to affect the bacteria E. coli. So bacteriophage is not a bacteria, it is a virus. You should know that. Then list the goals of Human Genome Project. These are the goals: identify all genes, this, determine the complete sequence, have the database, improve the tools, transfer the information, and address the ethical, legal and every issues over there. Now, mention the criteria for a molecule that acts as genetic material. So you should know what is the criteria. Okay, it should be able to generate its replica. It should be chemically and structurally stable. It should provide the scope for slow changes, my dear children. It should be able to express itself in the form of Mendelian characters. All these features are there, and it's been done by DNA. So DNA is the perfect genetic material because it is chemically and structurally stable than RNA. RNA has an extra OH group which makes it like an enzyme. So it is catalytic. It is not that stable. Okay. Should have slow changes. Obviously, for evolution you need it. Otherwise, how would we evolve better, right? It should be able to express itself, and it should be able to generate its replica. These 4 points you should have it in your head. Now list out the salient features of double helix model of the DNA. You should be knowing that DNA is made up of what? Pentose sugar, there is a nitrogen base, there will be phosphate group, everything. Now together you will call it as a nucleotide. Nucleotide joins with another nucleotide. What is this bond between the nitrogenous base and pentose sugar? It is N-glycosidic link. What is the bond between the 5th carbon and the phosphate group? It is phosphoester bond. What is the bond between the 3rd carbon and the phosphate? It is phosphoester bond. Together you have 2 phosphoester bond, you call it phosphodiester bond between the 3rd carbon of one pentose sugar and the 5th carbon of another pentose sugar. You get it? Now if we talk about the DNA, if the whole structure of the DNA is like this. So this from here to here, this is one pitch of DNA, my dear children. It is almost around 34 Armstrong in length. Okay, 34 Armstrong, that means 3.4 nanometer. 34 Armstrong means 3.4 nanometer, and over here you will be having 10 base pairs. How many base pairs? 10 base pairs. And in between each of these bases, what is the distance between them? It is 3.4 Armstrong. 3.4 Armstrong is the distance, that means 0.34 nanometer. So that is the distance between each bases. All this information is really important. How many base pairs you have? You have adenine and guanine, and you have cytosine and thymine. Okay, in the case of DNA, adenine and guanine are purines. Okay, thymine and cytosine are pyrimidines. In the case of RNA, you have one more, that is uracil, my dear children. You do not have thymine, you have uracil in the pyrimidine sector. And the DNA is running anti-parallel. It is very important, anti-parallel to each other. One is in 5' to 3' direction, other one is in 3' to 5' direction. Anti-parallel, please do not forget it. Okay, now DNA replication is fast, accurate, energetically expensive, substrate and enzyme dependent, initiated from a specific site, cannot uncoil on its entire length, everything. How do we explain this? by explaining DNA replication. So you talk about all the steps one by one, that it starts from origin of replication, that means it has a particular site for initiation, as it was saying, okay. Helicase unwinds it, so it is enzyme dependent, and then you have single stranded binding proteins so that they do not coil back to each other. There is primase, another enzyme adding primers, okay. Elongation, DNA polymerase elongates it, and it is substrate dependent because it needs nucleotides as a substrate. There is leading strand whether synthesis is continuous, lagging strand whether it's discontinuous. You find Okazaki fragments. Now to join this thing, okay, discontinuous thing, you have DNA ligase. Now it is accurate. Why? Because DNA polymerase does the proofreading as well, whether any mismatch is there. Termination happens when the replication fork which is there, it meets each other. All these things when you explain, you will be able to explain the other things which are included over here. Okay, now getting ahead. Given below sequence of nucleotides in a particular mRNA and amino acids coded by it. So you have, when basically this is given, you have to tell about the genetic codes features. Okay, so AUG has a dual function. It codes for methionine. It's also a start codon. UC. Basically, there are two codons for one amino acid. That means this codon is degenerate. Okay, that is the feature, that is degenerate feature. You see any full stop, comma, any punctuation? No. So this is contiguous. Okay, and now one codon codes for one amino acid only, not for any other amino acid, so it is unambiguous. Okay, UAA does not code for anything, that means it is a stop codon. All the codons that you see are in 3-3 format, so it is triplet. How many codons do you have? 64 codons. You have 61 of them codes for amino acid, 3 of them do not code for anything. You get it? So all these points you have to write it down properly. If you are writing it, you get marks, and they are universal, my dear children, remember that also. So phenyl, UUU codes for phenylalanas and also in the bacteria. Okay, in some it might not code the same, but yes. Exceptions are given, so that is mitochondrial codons and the protozoan codons, in which it could be different than us. Okay, now describe the steps involved in DNA fingerprinting technique, just like how you were talking about VNTR, right? You're talking about VNTR, you have to explain those steps only in DNA fingerprinting also. So DNA is extracted from the hair, blood, saliva and things like that, then it is digested, then it is isolation of DNA happens, then digestion happens, then separation happens, then transfer to the membrane happens, then DNA probes will come and attach, and it is visualized using what? And the X-ray or UV light and anything which is there. And my energy is like okay. So yes, we are doing this for you. Are you doing anything for us? Are you studying for us, or are you not doing that? Very bad if you are not doing that. So, and if you are doing that, so answer the following. Construct a complete transcription unit with promoter terminator on the basis of hypothetical template strand. So this is a template strand given over here. If you know, you know. So in DNA there is one template strand, another one will be coding strand. The one that undergoes transcription is known as template strand, so it will be in the direction 3' and 5'. So always when the arrow goes this side, that means it is 3' and this is what? 5' direction, my dear children. Okay, now you have to construct both in a hypothetical transcription unit. Sorry, not in a hypothetical, you have to make a transcription unit, and then you have to transcribe it and show the mRNA also. So how do you make that? You just put down over here, this is the template strand, and over here the opposite complementary strand will be what? The coding strand. Okay, the coding strand is present at the 5' promoter sequence which is there, onto which the RNA polymerase will come and attach, will be present on the 5' end of the coding strand. Terminated region will be present on the 3' end of the coding strand, my dear children. Okay, got it? Now over here, when this particular thing is going to get transcribed, it is going to get transcribed into mRNA. So whenever you make mRNA, make sure you're writing in 5' to 3' direction, because transcription happens in 5' to 3', translation happens in 5' to 3'. Okay, and what? DNA polymerization, when DNA is being replicated, it also happens in 5' to 3', all 5' to 3'. Okay, so basically over here, how do you make the mRNA? So you have to take this strand and you have to make the complementary RNA strand, not the DNA. So this is DNA strand that you made initially. You have to make an RNA strand. So what do? Wherever there is T, you put U. Wherever there is T, you put U, and then you can write it down as it is. Okay, that's how you can make it. The salient features of human genome, so you have to list all of it. That is, human genome consists of 64.7 million base pairs. Then average gene size is this, that is, it contains 300 base pairs. But then you have dystrophin, which has 2.4 million bases. The total number of genes is this, 30,000. Okay, the functions of over 50% genes are not known. Less than 2% of the genome actually codes for protein, and then there are repetitive sequences which do not actually have direct coding functions. Chromosomal chromosome number 1 has the most genes, that is 2968, and chromosome number Y has the fewest, that is 231. Okay, scientists have identified about 1.4 million locations where single base DNA differences has been taken place. So all this information you have to write down. Okay, now go ahead, describe lac operon concept of gene regulation. So over here you have to make this diagram and then explain. Okay, so there is promoter sequence for a regulatory gene, there is promoter sequence for the structural gene over here where Z, Y and A is there. Z codes for beta galactosidase, Y for permease and A for transacetylase. Okay, and there is repressor protein that is being made, it binds on the operator region, which is why the RNA polymerase which binds over here would not be able to move forward unless and until there is an inducer, that is lactose or allolactose, which comes and binds to the repressor. Now the repressor is not free to go and bind there, hence transcription will be happening. Beta galactosidase will be breaking down lactose now. Okay, permease would be letting it enter and transacetylase phase do the other functions. So all these points you have to write down as well in switch off condition, as well as when the lac operon is turned on. How does it turn on? With the help of lactose or allolactose, and tell what are the enzymes being produced, what are their functions. Over here you can just read through. If I go on explaining, it might take a huge lot of time. Okay, so these are the things. Now DNA replication is said to be semi-conservative. Why? Describe the experimental proof of Michelson and Stahl to show DNA replication is semi-conservative. So why is DNA semi-conservative, replication semi-conservative? So if you have two strands, okay, and now when these two strands are going to replicate, you are having the parental strands, from here you are going to get new strands. So parental strand is still being conserved and you are getting new strand, that is why it is in the semi-conservative. Okay, there is half parental, half new. Fine, and over here you have to show the experiment, that is Michelson and Stahl experiment. What they did, they basically grew the E. coli in a medium that had N15 isotope, that is a heavy isotope. So both the DNA strands and nitrogen is found in DNA, so it will be taken up. So both the strand had this particular N15, so it will be in heavy zone. When it is put in cesium chloride and sedimented, my dear children, it will be heavy, so it will be sedimenting down. Now if you are putting the same thing, E. coli, into next medium that is having N14, okay, what will happen? Now the new strand which will be formed, it will be having N14, the older strand will be having N15. So this time it won't be settling towards down, but it will be in hybrid condition, because one strand is N14, other strand is N15. So it is hybrid. Again, if you keep it for 20 minutes, because 20 minutes is the replication time for E. coli, it divides every 20 minutes. So this is 20 plus 20, 40 minutes after that. If you see, my dear children, again this will divide into two strand, and again new strand will be formed, that will be 14, 14, 14. So this time you will be finding two hybrid strands and there will be two light strands. Okay, both of them are from N14 itself. So this is how he showed that DNA is replicating semi-conservatively, because every time there is one parental strand and there is one new strand added to it, which is why the red one is still there in every generation. Get it? Ouch. Cool. So it was done by Michelson and Stahl with the help of E. coli. Now, so this is the whole thing written, you can just go through it. Now Oswald Avery and others have continued Griffith's transforming principle to prove DNA as genetic material substantiate. So for here you have to explain that if you use, if you have heat killed S strain and if you are putting live R strain and you are putting protease, still transformation still will be taking place. That is, the live R strain is going to convert into live S strain and it is going to cause the trouble. Now heat killed S strain, if you are going to treat with RNAs and then put it with live R strain, still transformation will be taking place. But if you are going to treat this thing with DNAs, the DNA is going to get ruptured over here. Okay, so what will happen? The live R strain is not going to convert into S strain. So there is no transformation that is going to take place. So with this they concluded that yes, DNA is the genetic material, because when DNase is being put, that's when transformation is not taking place. So probably whenever messing up happens around the DNA, that's when the transformation would not be taking place. Cool? Yes. Now explain the process of translation in protein synthesis. So you have to talk about translation. The first thing that happens is the tRNA which is there, it will always bind with the amino acid on the 3' end. So tRNA is like this, it is an RNA, it has 5' and 3' end. On the 3' end, okay, what will happen? The amino acid will come and bind. This is known as amino acylation or charging of tRNA. That is the first thing that happens. Later what happens? Okay, so I will draw and show. First thing, over here you have 5', here 3', here amino acid comes and attach. This is known as charging of tRNA. This happens in the first phase. Now you have the mRNA strand. mRNA strand will be binding with the smaller subunit. Then the tRNA containing the initiator codon, initiator anticodon will be coming and binding. So here initiator codon is AUG, the anticodon will be what? U, A and C. and here it will be methionine. It will come and bind. Then later what will happen, that larger subunit will also come and bind. Slowly that ribosome which is there, it will move throughout the entire mRNA in 5' to 3' direction. That's how things are being formed. So activation or charging of amino acid, that is being done first. Then initiation, where mRNA binds to small ribosomal unit, the start codon is recognized by initiated tRNA, and the large ribosomal unit also assembles. Now elongation, where tRNA brings amino acid to the ribosome, matching codons on mRNA through its anticodon. Peptide bond formation occurs between the two tRNA, my dear children. Okay. Now ribosomes move along the mRNA in 5' to 3' direction. Now termination, when a stop codon, that is UAA, UAG, UGA is reached, translation stops as the release factors binds to it. In the stop codon there is no tRNA that will come and bind. It is the release factor that will come and bind, that will basically stop the whole translation process. So write that. Okay. Now describe Hershey and Chase experiment. So here you have to talk about the bacteriophage, which is labeled by S35, okay, and S35 it will become a part of what, the protein capsid, not the genetic material. Over here the radioactive phosphorus, that is P32, it will bind to the genetic material. Now over here you find at the end that after blending, after centrifugation, everything, the cells are are radioactive, but over here the pellet which is there, not the pellet, the supernatant which is there, my dear children, that is radioactive. That means the cell has not taken up. That means proteins have not been passed down. Okay, but over here DNA is being passed down, which is why it is readily taken up in the cells. So all this experiment, bacteriophage is a virus and this is E. coli. You have to show properly, remember S35, 5 looks like S, you can remember like that, that is radioactive. Ouch. So this is the whole thing in the, you can read it, you can write it like this, labeling of the phages, then infection, then blending, then centrifugation, observation that you find out. Now after this we have evolution, my dear children. Evolution, as I said, is an important chapter, it's a 7-mark chapter. So what are the questions? Potato tuber and sweet potato tubers are result of convergent evolution and divergent evolution, homologous, analogous organs, very much important. So convergent evolution, that means analogous organs are there, A, C, you remember like that, and HD, homologous organs and divergent evolution. Okay. Now why are they showing convergent evolution? As in both potato tuber and sweet potato are not related to each other, but still they are converging to a point that they have same function, they are doing the same function, that is to store what? Extra food, that is starch over there. But potato is formed from the stem and sweet potato is formed from the root. So both of them do not have a common origin or a common structure, but structural difference, okay, but they would be having a common function, which is why we are seeing the converging, they are showing convergent evolution, and these organs are known as analogous organs, my dear children. So different origins. Okay, why? Because it is modified stem, this is modified root. Similar function because both of them are storing nutrients. And independent evolution, these adaptation evolved separately in unrelated species to do the same purpose, which is why it is convergent evolution. And these organs, as I said, it is analogous organs. Now write a short note on Neanderthal man. Neanderthal man came towards the end, my dear children. It is about 1 lakh to 70,000 years ago, that's when they were seen. Now it was they have a brain capacity of 1400 cc cranial capacity, that is really great. They were the first ones to protect their body and bury their dead. So the certain amount of emotions ignited in them, my dear children, and they are known as Neanderthal man. All this, the origin of man is very much important. You should know all the stages. Okay, the question would be coming upon, I will be showing you that. Now mention the evolutionary significance of the following organisms, shrews and lobfin, a fixed question every time it comes. So shrews is kind of a connecting link, or basically it gives an evolutionary history between reptiles and mammals. So shrews are amongst the earliest mammals and represent the primitive ancestors of the modern mammals. So they are the primitive or the earliest mammals, and they are going to represent the primitive ancestors of modern mammals which are there. Now lobfins, which is there, they mark a critical step in the vertebrate evolution, where fishes were there. In this fish that is lobfin or coelacanth, it is also known as coelacanth. Okay, so what happened is that they were thought to be extinct. They had big fins and they could walk around with these fins on the land as well. Now that was very unique. Fishes could not do that. So they were kind of thought to be the ones through which the evolution of amphibians had taken place. Amphibians are the one that live both on land as well as water. So frog and everyone. Because these animals, that is lobfins, evolved in the first amphibians that lived on both land and water. They were the ancestors of modern day frogs and the salamanders. Okay, cool. So these things you should be knowing. Very important question. Now what are analogous organs? I told you. Analogous organs are different in structure and origin but perform common function. For example, wings of birds and wings of butterfly. Okay, both the wings are not same, but both are used for flying. Eye of octopus and mammals totally different, but both of them are used for seeing. Flippers of penguin and dolphin. Penguin is a bird, dolphin is a mammal. Flippers are used for swimming. Right. Now, root modification in sweet potato and stem modification in potato. Again, both of them are storing, but origin is different. So, all of them are analogous organs. Now, enumerate on convergent and divergent evolution with suitable example. Convergent, I already told about you about that. Okay. Now, divergent is basically they have the same structure and everything, but because they have moved to different areas, they are doing different functions with the help of that. For example, you have your forelimbs. So, forelimbs of us human beings, forelimbs of cheetah. We use it for moving around, doing exercise, dancing and doing everything like that. Now, over there in the case of cheetah, they do not dance, they do not do anything like that, they do not eat food like this. So, basically it is used for running and things like that. But if you see the structure, they also have humerus, they also have radius, they also have ulna, they also have carpals, metacarpals, phalanges, everything. So, structurally we are very same, but we have diverged to different habitats, which is why we have different functions. So, the same structure developed along different direction due to adaptation to different needs is called divergent evolution, and these structures are called as homologous organs. Now, over here my dear children, thorn of bougainvillea and tendril of cucurbita is also an example. Now, thorn is a thorn, tendril is a tendril which is used to coil around. Okay, both are evolving from the axillary bud. Both are going to evolve from the axillary bud, my dear children, which is why it is said that they are showing divergent evolution. They are moving to separate things because they are separating in this, they are living in the separate habitat. Cool. Now, write the salient features of the following human ancestors. I told you this will be coming origin of man. So, Dryopithecus. So, listen to me very much carefully. Okay, we had Dryopithecus and Ramapithecus. Dryopithecus were more ape-like and Ramapithecus were man-like, but they were very like so many years back. If you go like that, that's where you find it. Now, from Ramapithecus came Australopithecus. Okay, they were the tool makers and everything. Okay, they were found in Tanzania and things like that. Talking about Homo habilis, which evolved from Australopithecus. So, you got the first hominid-like features in Homo habilis. Okay, the first hominid-like features. They used to eat, fruits and all. They were tool makers again. They used to eat fruits. They are not meat eaters. Now, from Homo habilis came Homo erectus, who started eating meat and all, and the brain capacity was also increasing day by day. From here and all, you in the book the brain capacity has been mentioned. I will be showing up, right? So, Homo erectus is the one that had good amount of brain capacity. Now, they were also known as Java men because they moved around to China and regions like that. In Java, you know, in China there is a region like that. And from them came Neanderthal man and Homo sapiens. First came Neanderthal man, as I told you, they had 1400 cc brain carrier capacity and they used to cover their body, they used to bury their dead. And then from them, not from them, parallelly one more thing was happening, that is Homo sapiens, modern Homo sapiens, that is us, were also forming. Okay, so we also had good brain carrier capacity and things like that, but about us there is no other mention that is given in your NCERT. So, you have to write about all of them. So, how do you write about all of them? So, Australopithecus, my dear children, they were found 3 to 4 million years ago. Okay, and there is no much thing even in your NCERT. You can just write they were tool makers, they were from Tanzania and things like that. Homo habilis is the one that is given, and over here they were the first hominid. Okay, the first hominid, pure hominid-like features were there. 650 to 800 cc was their brain capacity. They ate fruits, they didn't eat meat, they were also tool makers just like their common ancestor, that is Australopithecus. Now, Homo erectus, which is there, it is having 900 cc. So, if we see 650 to 800 was there, here it has become 900 cc. They ate meat and they were known as Java men because they moved around to China and regions like that. Then from them you got Homo neanderthalensis, that is Homo Neanderthal man, my dear children, which came about 1 lakh to 70,000 years ago. 1400 cc was the brain, protected the body and buried the dead. From here only, okay, from erectus only, you got the next one. Where is that? Homo sapiens. So, it was found around 75,000. It is not 77 lakh 50,000. It is 75,000 to 10,000 years ago they were found. Okay, very recent. Now, it is like when they were evolving, some period in between that they were also evolving separately. Okay, now about primates over here. See, Dryopithecus and Ramapithecus were existing. They were hairy and walked like gorillas and chimpanzees. Ramapithecus were more man-like, whereas Dryopithecus was more ape-like. Ram, you can remember like that. Okay. So, they were like more man-like. Now, write a detailed note on adaptive radiation. What do you mean by adaptive radiation, my dear children? Adaptive radiation is basically when, for example, I'll make you understand the concept. Okay. So, over here, I have finches, Darwin's finches over here. Okay. So, these Darwin's finches moved to different areas. Okay, when they moved to different areas, they evolved separately. For example, there was a group of finch in which one finch had a beak like this. It happened because of mutation. But when they moved around to different area, they found that they were able to better adapt there. They were able to eat insect with the help of that particular beak. So they started eating insect. They started to eat grass. They started to eat something else and everything like that. So it is basically they moved to different areas. They got adapted there. You got it? They got adapted there, and that's how they evolved. This is an example of adaptive radiation. Now this was shown by not just finches but also Australian marsupials. Australian marsupials also went like that to different things. Okay, this was happening in what do you call it, Australian continent, whereas this one is happening in Galapagos Island. Okay, so in Australian continent this was happening, but apart from that, one more thing was happening. There were placental mammals. So there were Australian marsupials and placental mammals at the one point. They were both radiating. Okay, so what was happening? Both of them were radiating towards same site, and both of them were having same features. So what do you call when two uncommon ancestors are basically evolving and they are coming together to form something common? They have common function. This is known as convergent evolution. So when you talk about both of them together, you say that it's an example of convergent evolution. But when you talk about just one, when you talk about just Australian marsupials and they are moving from here to here, so this and this will be different from each other, but they are evolved from common ancestors. So this is divergent evolution. But when you have two ancestors or organisms over here and they are radiating together, they are two different separate things over there, but they are having the common function, which is why they are known as convergent, example of convergent evolution. I hope you got it. Yes. So I have explained all this thing, as I said, I have explained all this thing in a very detailed manner in all the 5 marker, 2 marker, 3 marker, Brahmastra series, everything. So please watch those videos, marathon series videos only you watch, that's more than enough. It is long, but it is very much important. Okay, with the help of a neatly labeled diagram, explain Stanley Miller's experiment in support of chemical evolution. So chemical evolution says with the help of inorganic things, they were able to make organic things. Now in this condition, the primitive earth, there was reduced atmosphere, that means there is no oxygen and the temperature was really high. So Stanley Miller also maintained something like this. So this was the experiment I did. He had boiling water, from here water vapor came in, and here you have electrodes which gives high amount of temperature, that is 800 degrees Celsius, my dear children, and you had the gases, that is methane, ammonia, H2 and H2. H2O and H2. Okay, all of them basically reacted to give down the organic compounds that was found over here, that is amino acids and nucleic acids and stuff like that. So you have to draw this and explain that. Remember, there is no oxygen over here, it's a reduced environment. So explain like this in point. Now state Hardy-Weinberg principle, explain any 4 factors affecting Hardy-Weinberg principle. Hardy-Weinberg principle says that basically what happens is that the gene frequency, the allele frequency would be remaining same generation after generation. For example, in a group, this much tall allele frequency is there, this much short allele frequency is there. It will remain same of generation after generation. Is that true? No. What if some other tall people came? This frequency will be changing. This is known as gene flow. Okay. Second thing, genetic drift. By chance, suppose earthquake came, tall people died off somehow, randomly. So again, gene frequency changes off. So this is genetic drift, where random changes in gene frequencies happens. Okay. Now mutation. If some mutation happened over here, there is something else that is being formed over here. Again, there is what gene frequency change. Then there is something that is known as genetic recombination. In sexual reproduction, genetic recombination happens where new combinations are being formed. Again, there is change in the genetic frequency over there. Apart from that, natural selection, only one type of organism is selected. Tall ones are selected because they are able to adapt better. So then what? Then again you are changing the allelic frequency over there. So these are the factors that affect the Hardy-Weinberg principle, and with that I remember one thing. In the side of Hardy-Weinberg principle only, they have given natural selection different disruptive, stabilizing and directional. So all these are really important if you are someone who is looking forward to MCQ questions from this chapter. Okay, I just thought of it, which is why I told you. So please remember all this very important 5 things that affects the Hardy-Weinberg principle. Going ahead, we have human health and diseases, my children, which carries 11 marks. So right there, let's see what is there. So draw the structure of antibody molecule, very repeated question. You should be making 2 heavy chains, 2 light chains, variable region, constant region, disulfide bonds between this and disulfide bonds over here. Here there is antigen binding site as well. Carboxylic end is here, amino end is over there. Okay. Going ahead, list 2 methods of HIV transmission: sexual contact, transmission of contaminated milk, sharing. not milk, contaminated blood. Okay, and now probably I'm hungry. Okay, now sharing infected needles from infected mother to the fetus through placenta. Only blood and blood gets mixed. Usually it is not happening, but if it happens, it happens. Now organ transplantation from an infected donor to the recipient, that can also lead to infection. Okay, name the drugs, its source and nature given below. So you should know all the name of the drugs. So over here, what is it? So over here, why is it not coming? When I wanted to come, arey. Okay, it's kind of playing hide and seek with me, so I cannot show. Can I show you like this? No. So Papaver somniferum is a source plant. Nature is depressant. What is this drug? This is heroin. Heroin is a depressant. Papaver somniferum gives two drugs: morphine, heroin. Morphine is what? A sedative and a painkiller. Okay, heroin is a depressant. Now cannabinoid source plant is Cannabis sativa. It affects the cardiovascular system. There is only one drug that affects the cardiovascular system. Please remember, it is cannabinoids. Ganja, marijuana, everything comes under this. Okay, cocaine is from Erythroxylum coca. What does it do? It affects the CNS, central nervous system. So central nervous system are affected by two things: opioids and cannabinoids. Opioids and coca alkaloids, that is cocaine and everything. Okay, cannabinoids affect the cardiovascular system. Nicotine basically increases the blood pressure and everything. That is something else. So these are the things that you should be knowing. And then going ahead, describe any three properties of cancerous cells. There is uncontrolled growth, that is metastasis. What do you mean by metastasis? Where basically the group of cells are moving from different areas. That is metastasis, that is found in cancerous cells. Now loss of contact inhibition. Usually what happens when one cell divides, another cell divides. When they come in contact, they stop dividing. But over here they will keep on dividing. This is known as loss of contact inhibition. They have lost that property. There is uncontrolled growth. These are the features of cancerous cells. Now name the pathogen, vector and symptom of malaria. Pathogen is Plasmodium vivax, P. malaria, and P. falciparum. Falciparum is the cause of the malignant version of malaria. Okay. Now vector is by biting of female Anopheles mosquito. Please remember, it is Anopheles mosquito. For chikungunya and dengue, it is Aedes mosquito. For filariasis, it is Culex mosquito. Okay, this can come in MCQ. Now symptoms is high fever and chill. Fever occurs on every alternate day. Vomiting also is there. Now differentiate between active and passive immunity. Active is basically your body is actively participating in this. You are giving the antigens and the body is producing antibodies. Okay, your body itself is producing antibodies. Passive is basically when the immunity is developed with the help of ready-made antibodies. For example, from the mother to the baby. With the help of milk, she is passing down IgA. Why? Immunoglobulin, because baby cannot produce it. So ready-made antibodies are given. That is passive immunity. Now active immunity, it is slow. It will take time because antibodies have to be produced. Passive immunity, it will be really fast. The immediate response will be there. Now active immunity, even though it is slow, it will stay in our body for a longer time. Okay, it provides lifelong immunity. Every time now this antigen comes, our body will fight against it. But this is going to be temporary. Why? Because the antibodies are provided for short time. Once it is over, it's over. Now memory cells are produced for long-term immunity. Obviously, in our body, memory cells are being produced when active immunity is there. In passive, it's not being produced. Now, what is allergy? Allergy is over-exaggerated immune response, my dear children. It is caused with the help of this particular antibody, that is IgE, allergy, and it is also because of mast cells. So, mast cells produce histamine and serotonin that causes vasodilation, swelling and everything. Ayyo. Now mention the types of carcinogens with example for each. So you have physical carcinogens like X-rays, gamma rays, non-ionizing radiations, like everything. Chemical carcinogens like tobacco smoke, biological carcinogens like oncogenic viruses which passes down the genes to us. Apart from that, in our body also we have proto-oncogenes which basically works normally. Okay, they produce normal things and everything. But once it is mutated, it is going to turn cancerous. Okay, that is proto-oncogenes. Cool. Now name the causative agents of the following diseases: malaria, Plasmodium; filariasis, Wuchereria. Okay, Wuchereria bancrofti, Wuchereria malaria; ascariasis, Ascaris; amoebiasis, Entamoeba histolytica; pneumonia, Streptococcus pneumonia; and Haemophilus influenzae. The question can be, you are given Haemophilus pneumonia. Do not get confused. It is Streptococcus pneumonia and Haemophilus influenzae. Make sure you are reading the whole thing properly. And now what are protozoan diseases? Only over here one protozoan disease is there, that is amoebiasis. Okay, ascariasis, filariasis is caused by Ascariasis. Plasmodium, say malaria is also there, that is protozoan diseases. Okay, this is protozoan. Both of them protozoan, both of them Ascariasis disease, and this is bacterial. Ouch. Now what are lymphoid organs? Write the functions of the following in response to immune system of our body. So over here, bone marrow, my dear children, bone marrow and thymus are primary lymphoid organs where the cells are being produced and where they get differentiate into antigen sensitive lymphocyte. They mature into antigen sensitive lymphocyte. Now the place where they are actually going to see the antigens, they are secondary lymphoid organs such as spleen, such as lymph nodes, such as tonsils, such as Peyer's patch. Now tonsil and Peyer's patch is associated with the mucosa layer. You know, the innermost layer is the mucosa layer, so it is known as mucosa associated lymphoid tissue. Under that comes tonsils and Peyer's patch. Okay, so you just go through all the things given over here. Bone marrow, what happens over here, thymus, what happens over here. If you're writing the points properly, you will get the marks. Okay, so thymus is where the T lymphocytes will mature also. Bone marrow is a place where the stem cells are there, it will produce everything. B cells mature over there, T cells come here for maturing, and then it goes to other areas. Please go through the answers, okay. Now explain different techniques which help in cancer detection and diagnosis. You have biopsy and histopathology, where you cut the cells, you cut the tissue and you test this, that is this is known as histopathology. Now imaging techniques are there, that is computed tomography, where X-rays is being used to generate a three-dimensional image of your internal organs. MRI, that is magnetic resonance imaging. Over here magnetic fields are being used and non-ionizing radiations are being used to get the pathological and physiological changes in the living tissue. Antibodies against cancer specific antigens are also used here. Okay, so if for example in our body the amount of antibodies are very high and we know that these antibodies are produced against cancer specific antigens, then we can be sure that this person has what? Cancer. And other techniques are also there in molecular biology where the genes are being detected. Okay, and you get to know whether that person is susceptible to get cancer or not. Now over here certain drug bottles are given and their function is given. Yeah, it is being shown and their function is given. Okay, now you have to find out what is the drug. So it is used by the doctors as a sedative and painkiller. There is only one, that is morphine. That is what? Morphine. Now helps patient to cope with insomnia and depression. You have two, one is barbiturates and other is amphetamines, again given in NCERT. Insomnia means you are not able to sleep for that. Depression, you know that. Okay, now increases blood pressure and the heart rate of consumer. This is nicotine. Okay, the people who smoke will be having this problem. Now acts as depressant and slows down body function. It is heroin. Heroin is a depressant, my dear children. Now affects the cardiovascular system. I told you there is only one that affects the cardiovascular system, that is cannabinoids, marijuana, everything. Now describe the measures taken to control alcohol and drug abuse. Avoiding undue peer pressure, educating and counseling, seeking help from parents and peers, looking for danger signs, seeking professional and medical help. You might feel that this answer is like, why should I study? But you should be knowing the points, otherwise you will not be able to frame it. So please make sure you're going through it. Okay. Now give the schematic representation of the replication of retroviruses, my dear children. So you have to mention that the virus is there which have a protein coat and the RNA, and it also has the enzymes. Okay, it also has its own enzymes. What will happen? It will come and affect the macrophage. It will send its RNA. Now with RNA is there, usually from our DNA RNA is made, and this is known as transcription. Now if you are doing the reverse thing of it, it is known as reverse transcription. It is done by one of its enzyme, that is reverse transcriptase. So it makes DNA out of it. Now this DNA is incorporated into our host genome. Now host genome's machinery is used to make the replica of the DNA and basically transport into mRNA also. Okay, to make the proteins out of it. So proteins are made, that is the capsid is made, the RNA is made, and now they move out from here and go affect the other cells. The HIV factory is macrophage, my dear children, but the cells that it mainly targets, it is T helper lymphocytes. Remember that, T helper lymphocytes. An MCQ question is coming. Okay. With that question is also done. Now we have microbes in human welfare, which is basically a 7-marker thing. Now here, what are biofertilizers? Biofertilizers are basically what? Organisms that are going to provide the plants with nutrients. So instead of putting fertilizer, which are chemicals, why cannot we just provide organisms which are going to fix nitrogen, phosphorus and things for them? Okay, so we have Rhizobium as a factor that provides nitrogen. Then we have mycorrhiza, which is a fungus and plant combination that gives what? Phosphorus. So these are going to act like biofertilizers. Sorry, the significance is what? It increases the nutrient quality of the soil. Now going ahead, define BOD, mention the commercial importance of Monascus purpureus. First thing, second question. Monascus purpureus is the one that produces statin, my dear children. Statin is there for stabilizing or lowering the blood cholesterol level. Okay, if we talk about BOD, what is BOD? Biochemical oxygen demand. Biochemical oxygen demand means the amount of oxygen required by organisms to break down organic matter. So basically, suppose there is an aerobic bacteria. It has some organic matter to break down. So aerobic bacteria, it needs too much amount of oxygen to break it down. So if it needs too much amount of oxygen, that means BOD is way too much. That means organic matter is also way too much. That is why it is needing that much oxygen. Now suppose organic matter is less. Organic matter is less, then aerobic bacteria will also be less. Okay, aerobic bacteria would not need too much amount of oxygen. So basically BOD will also be less. So BOD is the amount of oxygen required by aerobic bacteria, or basically bacteria, in 1 liter of water to break down the organic matter. Okay, if it is reduced, that means the water is free of pollutants, there is less amount of organic matter. So BOD refers to the amount of oxygen required to oxidize total organic matter by bacteria present in 1 liter of water. Okay, now complete the below given tabular diagram thing. Okay, let's see. So dash is the name of the microbe that produces lactic acid. That is lactic acid bacteria, that is lactobacillus. Now what is the use? It is used for basically converting the milk to curd. That is the use. Now you have methanogens. Name of the product. Name of the product is methane, and methane can be used as biogas along with other gases. Okay. Now you have E and F, but it is used for treatment of bacterial disease. If it is used for treatment of bacterial disease, my dear children, it should be an antibiotic. So it is penicillium notatum. It is penicillium notatum. It should come over here. Okay. Every time I do this mistake. So penicillium notatum, my dear children, should come over here. It is a fungus that produces penicillin. Penicillin is an antibiotic that is used for treatment of bacterial diseases. Cool. Done. Now explain the role of, okay, I will just put like this, otherwise you will say, ma'am, so this goes this side and this will be coming this side. Okay. Now explain the role of microbes in industrial products. So you should know. Okay, citric acid is produced by Aspergillus niger, which is a fungus. Acetobacter aceti produces acetic acid. Clostridium butyricum produces butyric acid. Lactobacillus produces lactic acid. Yeast produces ethanol. Okay, Streptococcus produces streptokinase. Streptokinase is a clot buster. It bursts the clot. Okay, Trichoderma polysporum produces cyclosporin A, which is immunosuppressant. It suppresses our immune system. Monascus purpureus produces statin, which lowers down the blood cholesterol. Okay, if you remember this thing, half of the chapter is essay done. Okay, now explain the role of microbes in household food products. So lactic acid, basically milk to curd conversion, and also for increasing vitamin B12. Baker's yeast is used to making bread, and toddy is a traditional drink obtained from fermentation of the sap. Large hole in the Swiss cheese is because of a bacteria that is Propionibacterium shermani that releases a lot of CO2, which is why that hole comes in. And Roquefort cheese is ripened by a fungus. Okay, so these are the things that microbes that is used in household. Explain the role of 3 organisms of biocontrol agents. Biocontrol agents, as in they can be used instead of pesticides, they can be used. So they would be eating down all the pathogens, all the organisms up there which can disturb the crop. So ladybird is one thing, a beetle which is there. It feeds on aphids and mosquito, my dear children. Then you have it is feeding on aphids actually, and there is dragonfly that feeds on the mosquito. You have Bacillus thuringiensis, which is used to control butterfly caterpillars and everything specially. In Bacillus thuringiensis, they have one gene that produces prototoxin. This toxin, when ingested by the insect larvae, what happens in them, it becomes active because it is alkaline pH in their gut. Okay, now what will happen basically, when this thing happens, alkaline pH is there in their gut, my dear children, there will be pores that will be made and they will die off. Okay, Trichoderma, as we studied, it is producing cyclosporin A that is used for humans, but Trichoderma has one more function. It is there in the root of the plants and it kills the other fungus which are there in the roots, okay, which are not good. Now baculoviruses are also there that attack insects again and other arthropods. Okay, now the majority of baculoviruses come in the genus Nucleopolyhedroviruses. Very important question. Now viruses are excellent candidates for this, that's all. So at least write this. Now what is the significance of BOD? I told you, if BOD is very much, that means organic matter is way too much, that means there is pollution. Okay, now give an example for fungus found in mycorrhiza. So it is mycorrhiza, in that mycorrhiza is a combination of the plant root as well as the fungus. Now it is not the example of fungus that is asked, it's the genus that is asked, and the genus is Glomus. Okay, what is it? Glomus. Cool. Now, with respect to the microbial products, its source and use, identify A, B and C in the following table. And also say what is BOD and flux. So cyclosporin A, I told you, it is produced by Trichoderma polysporum. It is a immunosuppressant. Monascus purpureus produces statin, that is a blood cholesterol lowering agent. Streptokinase is a clot buster produced by Streptococcus. Okay, so these are the things that you should be writing down. What are flocks and what is BOD? BOD I know, I explained you. Flocks I would be explaining you, okay, right away. Now what is a typical biogas plant? So here is where you are going to put dung and you are going to mix it with water. Here in the dung there will be so many methanobacteria, methanogens, okay. Now over here when they are transferred to this particular thing, this is anaerobic situation over here, there is no oxygen. So what will happen? These methanobacteria will be acting on the organic matter in there and producing what? Biogas, which can be taken through this biogas chamber to the homes for kitchen use or basically for electricity, whatever you want, and then whatever is left behind is put to the next thing, and from here it can be taken up for using as fertilizers or manure. Okay, so please make sure you're drawing it properly and writing it also. Now explain the stages of sea waste treatment. So there is primary treatment, there is secondary treatment. In the primary treatment, what do? You do filtration and sedimentation. Filtration as in the one that float, you remove it out. Sedimentation as in the one that is big, it will come down, you remove that out as well. That is filtration and sedimentation, my dear children. Apart from that, what do you do? Apart from that, basically what do you do? Okay, so then what is left is known as primary effluent. This primary effluent is treated in the next thing, that is secondary treatment. In the secondary treatment, in large aeration tanks, okay, aeration tanks, but there is air, aerobic bacteria is put. Now the aerobic bacteria will be feeding on the organic matter, okay, and at this point of time, as I said, BOD level will be way too much because it needs too much amount of oxygen. Now when it does so, the aerobic bacteria makes groups with fungal filaments and certain extracellular things that is there, and that is known as flocks. That is what flocks, my dear children. You have to imagine what I am saying. So they are eating on organic matter and things like that. After a point of time, every organic matter will be eaten up. How do you know that? Because the biochemical oxygen demand will be going down. Okay, that means they have eaten up everything. Now what is done is that whole thing is being transferred to the nest settling tank. The flock and everything will be settling down, and this is known as activated sludge now, and whatever is left behind, it can be removed into river and streams, whatever you want, because it is all cleansed now. Now this activated sludge, no, you take little part of it and you put it back into the aerobic tank, because another group of things are going to come to, so you need some inoculum, right? So it acts like that. Rest what is there, you are going to transfer it into an anaerobic sludge digester. Anaerobic sludge digester, that means where air is not there, where oxygen is not there, you are going to put it there. Now what will happen in this anaerobic sludge digester? There will be anaerobic bacteria, the one that does not need oxygen. They will be feeding on this aerobic bacteria and breaking down and killing it. You get it? You get it? Okay, they will be working on it, they will be feeding on it, they will be killing it completely, and that's when they produce certain biogas, that is methane, H2S, CO2, and that's how you get biogas as well. Okay, so flocks is nothing but a group of a mesh-like thing that is formed because of the aerobic bacteria, as well as the fungal filaments, as well as the extracellular thing that they are going to produce. Okay, so these are the answer for primary and secondary treatment. You can just go through it. Your ma'am is terribly tired, and this is the ecosystem, that is the 9th chapter that we were talking about. So over here again, represent diagrammatically the pyramid of number in terrestrial ecosystem. So over here, this pyramid of number will be always upright, my dear children, on terrestrial ecosystem, because the number of producer will be way too much. Okay, plants, trees and everything will be way more than tiger. Tiger will be 1 or 2, tree will be so much. Okay, then the rabbits will also be too much. Secondary consumer which is there, that will also be there, but the tertiary consumer will be less. So pyramid of number, you have to mention the numbers, you have to write producer, primary consumer, secondary consumer, tertiary consumer, everything. Okay, going ahead, yes, so you can explain it also, but you don't need to, because they have just said to explain like that. Now example of grazing food chain is given below. Grass is eaten by grasshopper, birds, and then man. So basically you are asked to make what? Energy thing. So you make grass and then you write grasshopper, write it properly, okay, not like me, and then you like birds, and then you write man and mention this is trophic level 1, this is trophic level 2, this is trophic level 3, this is producer. Okay, this is primary consumer, secondary consumer, tertiary consumer. So over here, pyramid of energy, if 10,000 kilojoules of energy is over here, 10% of this 10,000 is being transferred to the next, so it will be just 1,000, just 1,000. From here only 100, ayo, I meant 100, okay, 100 kilojoules over here. Now from 100 it will be just 10 kilojoules up over here. Cool, done. So this is what? This is the graphical representation, okay, of energy, pyramid of energy. Fine. You have to mention about that basically 10% is being passed on. Okay. Now pyramid of energy is always upright, can never be inverted. Give reasons. Why is it always upright? Because energy decreases at higher trophic levels. Energy is transferred from producer to consumer. At each step resulting in less energy. Okay, because of metabolism, they are doing their metabolism. So obviously when you go up, it will be less energy will be found. That is why it is always upright. Now inefficient energy transfer is inefficient, which is why 10% is lost. That is why obviously other one will be having less only. How can we expect it to be inverted? Now decreasing biomass and organisms. At higher trophic levels, there are fewer organisms compared to lower trophic levels. Yes, right, trees are more, tiger is less. This also reflects the decrease in energy as fewer organisms can be supported with the available. Obviously, you are losing lots of energy, just giving little amount of energy. How can you expect lots of organisms to be there up there? So that is why it is upright. Again, ecosystem stability. An upright pyramid supports ecological balance. An inverted one would destabilize the system. Okay. So these are the reasons why it is always upright. Put down the reasons properly. Now mention the functional component of ecosystem. Functional component was productivity, decomposition, energy flow, nutrient cycle. If this is not happening, the ecosystem cannot be maintained. So you have to talk a bit about all of this thing. Okay. Now what is trophic level? Trophic level is the level occupied by an organism in the ecosystem. That is trophic level. Refers to the position an organism occupies in a food chain or a food web based on its feeding relations. That is trophic level. Now ecological pyramids have limitation. Why these pyramids have limitations, my dear children? First thing, ecological pyramids focus on the producers and consumers but often forget the decomposers. They also play a major role, but they have not mentioned anywhere, right? Now ecological pyramids often represent a simplified linear food chain. But is it like that? No, we have a food web. Okay, tiger would not always say that I would eat only rabbit, not the deer. If it sees deer, it will go and eat the deer. So that is food web. Okay, there are different food chains happening together. So that is not being considered in the ecological pyramids. So these are things that you have to write. And the trophic level represents a functional level, not a species as such. A given species may occupy more than one trophic level. That is also there. One particular species can occupy more than one trophic level as well. It can be an omnivore. It can eat grass. It can eat another organism as well. Okay. So yes. Now explain the steps involved in the process of decomposition. So over here, my dear children, first thing you have to tell what is decomposition. It is a breakdown of complex organic matter into inorganic things. Now the steps. One is fragmentation, where the organism eats it, breaks it down. Okay, when the organism itself eats it, will be breaking it down into smaller particles. While doing so, what will happen? Certain soluble inorganic things will be moving. Okay, certain soluble things would be leaching down into the soil. That is known as leaching. It will be absorbed down into the soil and they won't be available now. Okay, now what happens? The organic matter which is left, you have to break it down. That is known as catabolism. Organic matter to inorganic matter, it is known as catabolism. Fragmentation means bigger things into smaller things. Okay. Now humidification is when you are breaking down organic into inorganic, there will be a stage where you are going to see a partially digested form. It is not fully digested into inorganic, it is partially digested thing, that is humus. So the formation of humus is known as humidification. Lastly, when the minerals are being taken up from this humus and everything when it is finally broken, it is known as mineralization. Okay. The humus further degraded by some microbes and release of inorganic nutrients occur by the process of mineralization. Now what are the factors that affects decomposition? Very important question. Okay, large amount of oxygen, high temperature and moist environment is required for decomposition. There should be large amount of oxygen, that for aerobic bacteria, high temperature and moist environment. This is a must for decomposition. You can get it as an MCQ question as well. Now, biotechnology, principle and processes, that is a 7-marker question, 7-marker chapter, my dear children. Now, this I have not included in the passing package one, but yes, you can read it if you are not sure about, if you are not able to understand the other chapters, then you can go ahead and obviously you should be studying the whole thing, right? So, over here, given below are the diagrams of plasmid A and B. You should be following plasmid A and B. So, this is a plasmid where, okay, plasmid B is the one where LAG-Z gene is there. LAG-Z gene codes for beta-galactosidase. So what happens usually is that if you put X-gal, this LAG-Z gene produces beta-galactosidase and this will convert the X-gal into blue colored thing, blue colored component, and it will show blue color. So if you are inserting something into this, our gene into this, our gene of interest into this, what will happen? It will now not produce the blue color colonies, it will be white color colonies. So in that way we can easily identify whether the organism has taken up this particular plasmid that have our gene of interest or not. Similarly over here, this is a plasmid that has ampicillin resistance and tetracycline resistance. Okay, again if I am putting it over here, putting my gene of interest over here, it won't be tetracycline resistant anymore, it will be just ampicillin resistant. Again this plasmid can also be used. So you are asked which one can you use? You can use both, but this one will be better because you can see color. Okay, now what is insertional inactivation? Insertional inactivation is when you are inserting something and that is why the pre-existing gene is not able to show its function, it is getting inactivated. Lac Z gene is not now going to produce beta galactosidase because it is totally inactivated. I have inserted something over there. Now will the number of culture plating you should make is same or different to select recombinant if insertional inactivation is possible? So culture plating is it different or same? So in Lac Z gene you can have single plate. Okay, there is a single plate, my dear children. In this you can have the E. coli. This is another E. coli. This E. coli has a plasmid which has our gene of interest. This E. coli has the older one. So what will happen if I put this color, just a second, okay. Over here the color will turn into blue because it has still the thing, but here it will be white colonies itself, so I can select this white colonies. It can happen in one culture plate. In the case of this ampicillin and tetracycline, what do? Basically first thing, I put it in ampicillin resistant thing. Okay, I put ampicillin antibiotic and I see whether it's growing or not. If both the E. coli are growing, that means both of them have taken up the plasmid for sure. That is why it is resistant to ampicillin. Now I have to check whether the plasmid has this particular thing or not. This plasmid has taken up the gene or not. For that what will I do? I am going to use the next plate in which I am going to treat it with tetracycline. So the one that has my gene of interest would not be resistant to this tetracycline, it won't be able to grow, but the other one will be able to grow. Okay, so I am going to choose what is able to grow. So there are two separate platings that are required over here. The answer is written. The two platings you can just go through it once. Okay, or you can see the 5-marker question. I have explained it to you in a very nice manner over there. Okay, in the marathon series. Now study the diagram given below and answer the question, that is what is EcoR1? EcoR1 is what? And restriction enzyme. It is a restriction endonuclease. That means the one that cuts inside the DNA. Okay, now how is the action of exonuclease different from that of endonuclease? Exonuclease cut the DNA from the end, endonuclease cut the DNA from the middle. Now how are sticky ends formed on a DNA strand? How or why are they called so? Why are they known as sticky ends? Because what do you do with the enzyme, my dear children, is that you have the enzyme EcoR1, no, it has a PUC one site, it has one restriction site, that is 5GAA-TTC. So it will be searching for 5GAA-TTC and going and getting marked. So over here there is GA, so there is 5GAA-TTC. So over here you are going to cut between G and A, here also you are going to cut between G and A. These are palindromic sequences which are read in same way both sides. Okay, similarly the foreign DNA which is there, it also will be cut like this between G and A, and here also between G and A. Now what you are going to do is that you are getting sticky ends. They are single stranded things which do not have any base pair. So they are going to be complementary to each other, they are going to bind to each other, and in that way I am able to join two separate DNA, vector DNA and the foreign DNA. So that is the use of sticky ends, my dear children. So you can write that as well. Now write 3 vector-free techniques that can be utilized to transfer recombinant DNA into a ready host cell. How do I transfer my DNA without a vector? That is without an organism basically. So there are 3 methods. One is electroporation, where I am making a pores with the help of heat swap treatment. Okay, I can create pores and there cesium chloride is being used. Okay, so over here with the help of the pores things are being inserted and the pore is being closed because of heat and let's say cold thing is there. Now apart from that, in the case of animals you have micro injection, where in the nucleus you are going to insert the DNA with the help of a micropipette which is there. That is micro injection that happens in only animals. Now in the case of plants, there is gene gun method or biolistic method, where you have gun because they have an extra cell wall, injection would not get in. Okay, so with the help of that there is gold and tungsten things onto which you wrap around the DNA and you send it. Okay, that is biolistic method. So these are the vector free methods and it has been written over here: gene gun, microinjection and electroporation. Now my dear children, there is agrobacterium tumefaciens which also acts as a natural vector for cloning genes in plants. How they can act as a natural vector? Basically they are bacteria and they cause tumor in the case of plants. Okay, because they have a separate type of plasmid which is known as Ti plasmid. tumor inducing plasmid. And this TI plasmid is very famous because it has a DNA that is tDNA, transfer DNA. So what does the bacteria do? It goes and affects the plant and it transfers the tDNA into the plant, causing tumor in the plant. You get it? That is the function of agrobacterium tumefaciens. So instead of having this DNA, tDNA, what if I put my gene of interest, still it will be going and infecting. In that way, it acts as a vector for plants. What about vector for animals? It is retroviruses, remember that. And a vector for animals, it is retroviruses. Vector for plant, it is what? Agrobacterium tumefaciens. Okay. Now, write any 4 tools used in recombinant DNA technology. Mention in 2 methods for introducing alien DNA into host cell. Name the stain used to visualize DNA fragments in gel electrophoresis. So, write any 4 tools. Basically, it is enzymes, vectors, host organism and bioreactors. These are the 4 tools that you need for recombinant DNA technology. Enzymes for cutting, vectors for transferring, host organism for having incorporated it, and bioreactor, which basically this is where the E. coli or basically any bacteria would be dividing it into different number and also giving the product that is needed, increasing its number basically. Now mention 2 methods, that is microinjection, bioelastic, whatever you want you can. Vector mediated, it can be heat shock method, disarmed pathogen vector, that is also there. Now stain that is used to visualize DNA, my dear children, it is ethidium bromide, and you cannot see it with your naked eyes. You have to have UV light. When the UV light falls, the ethidium bromide will give a bright orangish color. Okay, that is there. Now explain the process of denaturation, annealing and extension with respect to polymerase chain reaction, define plasmid. Now why a restriction enzyme called molecular scissors? So denaturation, you should be knowing all this thing. Denaturation is when the DNA is going to get separated into 2 fragments at high temperature, that is 94 degrees Celsius. Now annealing is when you are going to add primers to the DNA strand, because you need primers for DNA polymerase to extend it, right? So annealing is done when you add the primers. It happens in 55 degrees Celsius. Now extension is when the DNA polymerase will be extending the strand, and for that it needs Taq polymerase. DNA polymerase, the other name for it here, it is Taq polymerase, because you obtain this polymerase from Thermus aquaticus. You obtain this from Thermus aquaticus, which is a bacteria that can survive at high temperature. So its DNA polymerase enzyme can also survive at high temperature. You get it? So Taq polymerase enzyme is being used, and the DNTPs, deoxyribonucleotide triphosphates, are being used, okay, for for adding the substrate so that each nucleotide are coming there, right? And that's there for this. And apart from that, plasmid is the extracellular DNA that is there in the bacteria, my dear children, or the prokaryotes. They have their own origin of replication and things like that, so they will replicate. They don't have anything to do with the chromosomal DNA which is there. Now why are restriction enzyme known as molecular scissors? Because they are going to cut the molecules, that is DNA, RNA and things like that. Now draw a neatly labeled diagram of simple stirred tank bioreactor. Mention the sterile air, mention the flat bladed impeller, okay, which is there to mix the air. There is acid and pH control, steam for sterilization is there, foam breaker is there, everything is up there. This is known as simple stirred tank bioreactor. Now what do we have? We have a neatly labeled diagram of PBR 322, very important diagram. So over here there is ORI site, origin of replication for the plasmid, ROP, where that this is important protein for replication again. Here itself you have PVU2, that is a restriction enzymes site. So this is a restriction enzyme, its site is this, okay. Now there is tetracycline resistance site and ampicillin resistant gene. Over here you have 2 sites, one is BAMH1 site and one is SAL1 site. Okay, here in ampicillin again 2, PVU1 and PST1. Then EcoVar1, CLAV1 and HIND3 are also there. So everyone is 1, 1, 1, 1 except this. This is PVU2 and this is HIND3. This is what? HIND3. Please remember, this is a very important diagram. And with that we complete that as well, and we come with biotechnology and its application, carry 6 marks. Okay, so over here ELISA is one of the methods of molecular diagnosis. What is the principle? The principle is it works on antigen antibody interaction. Okay, so first we put antibody, we know the antibodies already which are being released by a person who is having HIV. Okay, so antibody is already there. You put the antigen now, basically you put the blood now which has the antigen. If the antigen comes and binds it, how do you know? We put another antibody which is linked with some enzyme, So this antibody will also come and bind. First, or you have ready-made antibodies, you put antigens. To understand whether the antigen has binded, you put another antibody. it will be having enzyme linked to it. Now you put a coloring agent, it will change the color. This shows that that person has HIV. So enzyme linked immunosorbent assay works on the principle of antigen antibody interaction. A specific antigen antibody binds to its corresponding counterpart. This interaction is detected using enzyme linked antibody and the substrate, which produces the color change. Okay. Now, why are meristems preferred in tissue culture? Meristems are preferred because they are free of viruses, and the most common meristems used is apical and axillary. So, at the tip of the plant which is there, no, that is apical, and axillary is basically the ones that is there in the branches, in the nodes. Okay. Now, how did the American company Eli Lilly use the knowledge of rDNA technology to produce human insulin? Basically, what they did was, they took the gene for A peptide, they took the gene for B peptide, put in 2 separate E. coli, okay. Using a vector, they transferred it to the E. coli, and the E. coli produced A gene, A protein, A polypeptide and B polypeptide separately. They joined it in the laboratory condition. This was being done. So, initially we were using cattle as well as pigs, okay, you should be knowing that also, cattle and pigs insulin, but it was very dangerous for us because some people were kind of allergic to it, and the problem with giving the whole gene of ours was, if you put the whole gene in one E. coli, C peptide is also being formed. C peptide is a part of pro-insulin, it is a not active form of insulin. So, we do not want C peptide. In our body, the C peptide is cut, A and B are joined with the help of disulfide bonds. But how can you do it in the bacteria? Bacteria is a prokaryote. So, basically you do not have any option. So, Eli Lilly came in and they said, let us put A gene here, let us put B gene over here, they both produce A and B separately, let us join in the laboratory condition by using disulfide bonds. So, this was done. So, remember, proinsulin is a hormone that is not active, insulin is the one that is active, mature, okay, and it does not have C. So, that has been written over here, you can just go through it. Now, one of the application of biotechnology is to get pest resistant plant, just justify the reference to Bt cotton. So, you have to talk about the cry genes. The cry genes are there in the Bt cotton. They are responsible for prototoxin. They will be active when insect has it, because insect gut, mid-gut of insect will be having basic alkaline pH. That is when the toxin gets activated, and this toxin will be cleaving the epithelial cells, the inner lining of the gut, and cleaving that and basically killing the insect. So, this can be used if this particular gene, okay, the same gene, the cry gene, if it is put in plant, the plant can do the same function, right? The plant, when it is eaten up by the organism will be dying, so the organism would come and eat. So, that can be done, okay. Now, how is ADA deficiency cured by gene therapy? ADA deficiency means adenosine deaminase is not being produced. So, how can you cure it? In some other way. Basically, what do? You take the lymphocytes of our body, okay, increase its number, and then you are going to put the gene, okay, the cDNA for this gene, that is adenosine deaminase, and then since it has it, we can put it back into the body, the lymphocytes. It will help us for some time, and again we have to do repeated infusions. This is how the gene therapy is being used. It is not a permanent cure, please remember it. For a permanent cure, what you have to do is take the gene from the bone marrow, okay, and then put in the embryonic cells only. In the embryonic stage, if it is treated, it is treated, otherwise it is not. So, there is bone marrow transplantation, there is enzyme replacement therapy, but all of them, they have a problem that they are not completely curative. Okay. So, then we use gene therapy, which at least gives some help, and then, as I said, the best one is doing it in the embryonic stage, but for that you should be diagnosing it really earlier. Now, explain 5 benefits of creating transgenic animals. So, you have for understanding the normal physiology and development of the gene. So, basically you put the gene into them and see what is this gene doing. Basically remove the gene from them and see what is this gene used to do, okay, or basically study the diseases. So, you have different diseases like cancer, Alzheimer's and things like that, cystic fibrosis. So, the treatment methods which can be applied for these diseases can be done on these organisms and can be found out. Biological products can be obtained from them. For example, in our case, alpha-1 antitrypsin is being produced, but if you want to increase its amount in more, we can basically insert the gene that produces it, and this can be used for treatment of emphysema, which is a respiratory disorder. Okay. Apart from that, basically this thing, what do you call it, Rosie, there was a transgenic cow. It was made to produce alpha-lactalbumin rich milk, the protein rich milk, so that can be used. Now, vaccine safety, chemical safety testing, for all of this thing, transgenic animals can be used. You can write those points. Okay. Now, what is RNA interference? How does RNA interference help to develop resistance in tobacco plants for nematode infection? The name of the nematode that affects the tobacco plant is Meloidica incognitia. And what do? Basically, it will have an mRNA that will code for a protein, right? So you are going to silence this mRNA. How do you silence this mRNA? Basically, you are going to have this particular organism, okay. This second. Okay, so basically what do? You know the gene and everything that codes for the mRNA, right? You're going to take this gene, you're going to introduce this particular gene into a plant with the help of a vector, that is Agrobacterium tumefaciens. Now the plant will produce a double-stranded RNA. Now when the animal comes and eats it, okay, the plant, what will happen? Double-stranded RNA will be transferred to the animal. Now there are two strands of the RNA. This is a double-stranded RNA, my dear children. And the organism has an mRNA which is soon going to transform into protein. From this double-stranded RNA that it has totally ingested right now, one RNA strand will come and bind over here, making the mRNA double-stranded. Now will it be able to undergo the process of transcription? No, it won't. Not transcription, translation. It won't be able to undergo translation, and hence it won't be able to make the protein. That's how you can have resistance. So this is known as RNA interference. Okay, and the whole step is being given up over here, identifying the nematode gene, using the Agrobacterium tumefaciens to insert the gene, okay, and then in production of double-stranded RNA in the plant, and the RNA interference in nematodes when they are finally eaten up by the nematode. Now we have organisms and population, which has 5 or 6 marks according to 2 different blueprints, and this time I have finally made it correct, so you don't have any confusion. So mention the population interaction, my dear children. This is this year's model paper question. A Bington tortoise and goats, they do competition because both of them are going to fight for the grass, and Bington tortoise is going to fail, goats are going to win the game. Tiger and deer, tiger feeds on it, so it's prey and predator relationship. So predator is tiger, deer is the prey. Sea anemone and clownfish, it is commensalism according to your NCERT, okay, because sea anemone provides protection to the clownfish, but in general actual sense it is mutualism. Why? Because even clownfish does some advantages, okay, gives some advantages. It feeds on like the pathogens on the surface, pathogens and things like that, provide some food and things like that. But since sea anemone has a bigger function to do, they are providing protection, so I will say the work that clownfish done is not being counted at all. Okay, probably that is why the NCERT might have written commensalism. So let's go with commensalism, where one organism is benefited, another organism is not harmed or benefited. Okay, now wasp laying eggs in the fig fruit, both of them are getting benefited. One is getting pollination, other is getting a safe place to lay the eggs, so mutualism. Cuscuta growing on the hedge plant, parasitism, because one organism is taking the benefit, other organism is getting harmed. Now predators are prudent and conduits in nature. Why? Prudence because they are very sure that I do not have to overexploit. If I overexploit, I would not have food later on. And basically they are very clever in that they only target weak animals because they are easy to capture, and they are also making sure that the community which is there, it is having just healthy organism which can breed and give more food to them. So this is why it is prudent. Why it is conduit? Conduit because it is acting as a passageway for energy transfer and matter transfer and everything. Okay, now to lessen the impact of predation, prey species have evolved different defensive methods in plants and animals. Support your answer by giving one example each for plant and animal. So over here are you. So this is the prey and predator and prudent and conduits point. You can just go through it, okay, once more, because I suppose you might need it. And defensive method, you can talk about monarch butterfly. It is highly distasteful to the predator, okay, and some species of insects and plants are cryptically colored, so again it is a defense mechanism, they are not shown up. And in plants you have thorns in acacia, cactus, and the acting as a morphological means of defense. Now Calotropis produces poisonous cardiac glycosides against the herbivores that eat it. So all these things are there. Again, as I said, just go through it once. If you want proper explanation, you can watch the marathon series. Works? Okay. Now explain mutualism with examples. You can talk about lichen, which is what? A thing between symbiotic association between algae and a fungi. Algae provides food, fungi provides nutrients. Mycorrhiza between roots of plant and a fungi again. Plant provides food, fungi provides nutrient, that is phosphorus. Plant and pollinating agents, and fig tree, wasp and everything comes under this, okay. Now what is commensalism? Explain with any 2 examples. So you have to talk about commensalism where one is one is benefited, another one is not harmed nor benefited. So you can talk about orchid growing on an epiphyte on the mango branch. Orchid is there for getting sunlight, but is not affecting the mango. Okay, clownfish living among the tentacles, it is providing protection. Okay, the sea anemone provides protection, clownfish has no benefit. Sea anemone has no other benefits. as according to NCERT, barnacles on the back of the whales, and cattle egret and the grazing cattle. Grazing cattle, when they move around, it gives around certain insects, the cattle egret feeds on it. Now, state Gauss's competitive exclusion principle. According to this, when there are closely related species and when they are competing for same resources, my dear children, they would not be able to coexist. They would not be able to coexist. The inferior one will be eliminated. Okay. So, that is the thing about competitive exclusion principle. Now, what are ectoparasites? The one that live outside, endoparasite, the one that live inside. What are the three parasitic adaptations? Loss of unnecessary sense organs, presence of adhesive organs, yeah, so that you can stick. Loss of digestive system, high reproductive capacity, so you can increase the number. Complex life cycle including one or two intermediate hosts. Vectors to facilitate parasitization in its primary host. Okay. Write any four factors that influence population density. One is natality, mortality, immigration and emigration. Natality and immigration increases. Natality means birth rate, immigration means entry of new organisms, mortality is death rate, and emigration means exit of organisms. Exponential growth curve is when there is no limitation to things, everything is growing exponentially. It is represented by dn by dt is equals to rn. R means intrinsic rate of natural increase, n is the population density, and over here it is the difference. Okay, according to time, what is the difference? Always remember, it is a J-shaped curve that you get at exponential growth curve. Okay, when you are talking about logistic growth curve, when there is a limiting factor, it is an S-shaped curve, you call it sigmoid curve. Okay, now what is population growth? Again, population growth is increase in the number of individuals in a population over time. Explain the factors. So it is natality, mortality, immigration and emigration. Biodiversity and conservation, but before that, my dear children, I don't know how I missed it, but there is brood parasitism. And sexual deceit. Okay, brood parasitism is when organism basically such as cuckoo go and lay egg in the crow's nest, and what will happen? Basically over here the crow will provide nourishment. Okay, so isn't this parasitism? Yes, because someone is taking benefit of it, and it should have been given to its own kid, but it is not being doing that. Okay, it is providing nourishment to the egg of the cuckoo because the cuckoo's egg resembles its own egg. Get it? So this is brood parasitism. And sexual deceit is when there's a type of orchid, okay, which looks like let's say one type of a pollinating agent, honeybee, which is there. So it comes and tries to copulate, but it pseudo copulates. It looks like the female version of it. So this is sexual deceit, where again it is like cheating the plant, cheating the organism to do pollination. Okay, so these two things also you have to just go through. The name was Ophrys, I think, of the plant, orchid. Now biodiversity and conservation. So over here, my dear children, last thing. So briefly explain the significance of David Tillman's long-term ecosystem experiments using outdoor plots. So over here you have to talk about how increased biodiversity basically enhances the productivity as well. So ecosystem with higher species diversity show greater primary productivity as different species utilize resources more efficiently and complement each other. Now it also leads to stability in the ecosystem. Diverse ecosystem are more stable and resilient to what? Disturbance like droughts or pests. So if you have diversity, obviously at least some of them will be surviving, if not others. Now resource utilization will also be better. All the resources will be utilized better if you have more biodiversity. Okay, so productivity is increased, stability is increased, resource utilization also better. Now name any four extinct organisms: dodo from Mauritius, quack from Africa, thylacine from Australia, Seleucid cow from Russia, and three subspecies of tiger, that is Bali, Java and Caspian. These are the extinct organisms. You have to remember all of their name. Define biodiversity. Write any two type of biodiversity. There is genetic biodiversity and there is species. Biodiversity is what? Variety of life form that is there on earth, whether it be plants or animals, microorganisms, whatever is there. Okay. Now over here, genetic means it refers to variations of gene within a species, different variations of wheat or rice. This is genetic variety. Species as in different varieties of species in a rainforest or things like that. Cool. Now what do you mean by endemism? Mention any two causes of biodiversity loss. Endemism is basically species confined to a specific region. They are not found anywhere else. Now biodiversity losses can be happen due to habitat loss and fragmentation. where they are staying, that particular area is itself cleared out, for example clearing of forest. Over exploitation of something, alien species invasion, as an I am bringing down different things and these things are going to feed on my organisms or basically take resources from them, that. And co-extinction, if one is getting extinct, the other organism which is dependent on it can also become extinct. So these are the things for biodiversity losses. So give any 2 examples of sacred groves in India. Very important question. Khasi and Jaintia Hill in Meghalaya, Aravalli Hills of Rajasthan, Western Ghats of Karnataka, and Sarguja, Chanda and Bastar area of Madhya Pradesh. You will be asked which one are the sacred groves found in Madhya Pradesh, or which one are the sacred groves found in Karnataka. So you should be knowing about all these things, especially Meghalaya. Now, there are many reasons for conserving biodiversity. Briefly explain the reason for conserving biodiversity from narrowly utilitarian point of view. So according to narrowly utilitarian point of view, it says that we should be conserving biodiversity because it provides us with products such as food, such as medicine, industrial, and also because of the economic value. Okay. So these are very important things. Now in your NCERT, one thing is written about Khasi and Jaintia in Meghalaya, which is very much important. If you just go to the last page, you will be able to find it. Can you tell me what is it in the comment section? It is very important for your exam as well. Now, yes. Now what is ex situ conservation? Mentioned to ex situ means you are conserving them outside their natural habitat, such as zoological parks, such as botanical gardens, such as wildlife safari parks, and the conservation of gametes by cryopreservation, and also the seed banks and everything that you are having. Okay. So that is ex situ conservation. Going ahead, mention the 4 evil quadrate which causes depletion of biodiversity. Among vertebrates, which group of animals has higher number in global biodiversity? Among vertebrates, if you are talking, it is fishes that has the highest number of global biodiversity. If you are talking about invertebrates, it is insects. Okay. Now you asked about evil quadrate. That means 4 things. Okay, that is reason for depletion of biodiversity. I told you it is habitat loss and fragmentation, over exploitation, alien species invasion. For everything you have examples, Ecornia, and then Clarius garipinus that was taking down the our own catfishes which were there. So all examples you have to put it down. Okay, otherwise it is not an addition to your answer. Extinction, when one species become extinct, depend on it, also other species will become extinct. That is, for example, as a host plant, there is another pollinator. Both of them, if they are staying together, if one gets lost, other will also die. So these are things. Now, as I said, fishes have the most number. Now, alien species invasion causes decline. I told you all the things. The Nile perch. Okay, so these are the examples. Okay, of alien species invasion. So you have African catfish, that is Clarius garipinus. For aquaculture, you brought it, but then it was a potential threat to indigenous catfishes which were there. So this is what? This is a invasive species which was there. Now, carrot grass, Parthenium, lantana, and water hyacinth, they are all plants and they are all invasive species of plants which are not allowing the normal plants to live. Nile perch is there, it's a big fish. Okay, it is basically, it was introduced into Lake Victoria in East Africa, which led to extinction of ecologically unique assemblage of more than 200 species of native African cichlid fishes. So cichlid fishes were lost because of this particular thing. Done? Okay, so these are the things that you should definitely know. And with this, we complete the passing package, my dear children. So it has been long, yes, but definitely very useful for you. So I hope you go through each and every question up over here. Please do not ask me, are these questions fixed? There are higher chances that these questions could come because they are the most repeated questions, and by the term most repeated, that means they could be repeated every now and then. So make sure you are making your concepts really strong around these topics, and also you can read your NCERT. Right, obviously you will be reading your NCERT, it's for exams, you are doing it, right? So make sure you're doing that, and if you think that this particular video was helpful, do not forget to comment down. Lots of love to you all. You are going to do amazingly good. Bye bye, take care.